Good morning, everybody. Thank you very much for making it here. I just wanted to give you a sort of a brief history. This has been a labor of love, and we've all been working on it for about a year and a half. And I would like to start by giving profound thanks to my team. They're all here, and honestly, without them, this would not have been possible. And I'm privileged to be here and talk about the program, but the reality is that putting it together took a lot of effort, and only a small fraction of it was mine. So I would like to acknowledge the help that I had from Paul, Adrian, Jason, Matt. They're my CETAs. Jen Myers is my business finance manager. Kurt Yeager, he's a neurosurgeon, and he was the one advising us on TA1. The TA1. Gina Perry, she is the contracting officer. We are all going to be here and happy to take questions and basically mingle with you for the rest of the day. As you may have been aware, so we put a request for information. That was in March of last year, trying to understand the landscape and what the unmet needs are. We sort of boiled it down and had a workshop on this topic in DC in about June of last year, and narrowed down the program to the two technical areas that you are seeing right now. And it's been a long time coming. If I had a child at the time this started, it would have been biting by now. But you know, the good thing it's out here. I think there is a lot of excitement around the program, and we're really happy to see the interest that we have. And I don't want to take too much time. We have a long day ahead of us, and you'll be listening to us talking all day long. So let's get the show on the road. Jen will be emceeing this, and she will be introducing the speakers coming next. So thank you all, and enjoy the day.
Good morning, everybody. I'm Jennifer Myers. I'm Ilana's business finance manager, and I'd like to introduce Tyler Best, our acting director.
I'd like to maybe just start with a couple of questions. How many of you all, and I know people online can't raise your hand, but you can do that hand raise thing on virtually. How many of you have worked with an ARPA, whether that's DARPA, IARPA, ARPA-E, in the past? Okay, so a good third, maybe, here in the room. In particular, how many ARPA-H? Okay, so a good handful as well. That's good to know. That really helps level set to identify where we are and what's going on. For those of you who haven't worked with an ARPA, it's a little bit of a history here. ARPA-H is modeled after DARPA, the Defense Advanced Research Projects Agency, which was started in the late 50s in response to Sputnik, this little ball with the spindles coming off there. That was Sputnik, that satellite that was launched by Russia back in the late 50s. It was a big surprise to the US government, and DARPA was launched in response to that to prevent strategic surprise for the Department of Defense. Over the years, DARPA has developed many things. There's a diagram there of the first interconnected computers, the ARPANET, which later became the internet. You see the image of the stealth fighter and self-driving cars, et cetera, et cetera, et cetera. All of those developments that DARPA did, among many others, are what we want to do for healthcare, health. We want to take on the big risk, high reward challenges in healthcare. When I used to work in a lab, I studied biophysics of neurons. And if you look at the action potential, the firing of an action potential, if you take the derivative of that action potential, you see the dramatic change in slope. And that inflection point, that derivative, is where the action potential threshold is. That is what we want to do for health. We want to change the trajectory of health, the voltage of a neuron, and get us to fire. So if we take that analogy, we want to do that for health. We want to create those inflection points, those changes that dramatically accelerate what is happening for health and provide health outcomes that wouldn't be achieved otherwise. How do we do that? We, like all other ARPAs, we are a very unique funding agency. We are problem-based. We focus on solving the big problems in the community, in the healthcare system, in the biology, et cetera, et cetera. And we are seeking that radical change, not just that incremental slope of progress. We want to induce that inflection point. We have a lot of autonomy. We have a lot of freedom to operate. We use primarily other transactions, not grants. And so if you're expecting to get money via a grant and walk away and come back a couple of years later and tell us what you've done, if you've ever worked with Alana, you'll know that that's radically outside of the scope of what an expectation is. You're going to have regular check-ins. And if you don't meet the milestones and if you don't produce and progress, there will be some discussions on whether or not you're going to continue on. Alana, myself, other program managers are term limited. We are here for a 3-year term with a potential to renew for a second 3 years. That enforces and induces change so we don't become stagnant, so that we bring in fresh ideas, fresh approaches to what we're doing. Even this AIR program, Alana may not be around at the end of this program when it wraps up and finishes. So that may be transitioned to somebody else. Yeah, that's a little shocking even to Alana. Yeah, so you know, this you really have to demonstrate progress, achievement all along the way to continue that funding. And there's some slides describing that later. We don't have to necessarily go through that. Our ecosystem, unlike the Department of Defense and DARPA, where their ecosystem was mainly focused on that defense industrial complex, everybody needs healthcare. Everybody needs to be healthy. And so our customers are us. We all, Alana has a story of why she came to ARPA-H based on some personal experience and wanted to solve some things. I've had family members die of cancer. I'm sure everyone in this room has been touched by healthcare problems, regardless of where you're at. Our performer teams, we call the people that do the work, that do the research, the performers. That's you all. You come from academia, industry, private sector, et cetera, et cetera. Our stakeholders are you, the general public, the healthcare industry, insurance companies, doctors, nurses, pharmaceutical companies, et cetera, et cetera. We all work together to try and accelerate and improve that healthcare system. The ARPA-H model, as Alana kind of alluded to a year and a half ago, more or less, she was planning and initiate this concept that surrounds AIR. She held a workshop investigating what are the major problems in this space, and that workshop helped her team refine where the problem is. And that's the fundamental thing that the ARPAs do. They work hard to refine and define the problem and that problem space so that we're not going after those incremental solutions. She sent out a solicitation this past month ago, I guess it was now, where you're all interested in finding out more what you can do to progress. As you write everything up, compile all of the information, the projects you're going to do, that's sent in to Alana and her team. She evaluates that with subject matter experts, other government personnel that review those proposals. She deliberates with her team, narrows things down, makes some selection recommendations. She comes to me discussing what her portfolio for the AIR program, what she wants it to look like. I ask her specific detailed questions on metrics, who's going to be successful, why she thinks her selections will be successful or not. We go over that in detail. Then I sign off on that. It gets agency director approval. We start negotiating on your statements of work, on your budgets. And then a few months from now, your awards will be made and you jump in and start working. That's where we migrate to this performance state. I've alluded to this, sorry about that, before, about ensuring that you're meeting milestones and metrics and progressing. Or if you're not, let her know immediately so that you can pivot and adjust to what will work. Why you think the original plan isn't going to work, and adjust that quickly and rapidly so that you're not dragging your feet and you can move on. At the end of the day, she is expecting deliverables, demonstrations, products that will eventually go into the hands of doctors to treat stroke, or the microbot to treat whatever it is that you've designed that microbot to do. And at the end of the day, it's a demonstration, it's a product that will be delivered out in the hands of the people. And that model follows pretty much for all ARPA-H programs and efforts that we do. This is kind of the diagram of you all as performer teams. Some of you will realize early on that your effort just isn't going to succeed. No matter how much you want to pivot and shift the direction, what you had thought was going to be promising, maybe it was really, really risky and we want to take on that risk, but it just turned out that you couldn't prove that whatever it was that you're proposing is possible. And therefore you come to a mutual decision to end your work, and then you can move on and focus on other things. And that may happen at various stages along the duration of the AIR program. Or if you're wildly successful, you keep on moving, keep hitting those milestones, and maybe you get additional funds to do some more work because you've been so successful. And at the end of the day, your design, your product is going out into the market. That's kind of the summary of how the program process, et cetera, works. We have within ARPA-H 5 primary areas or offices that provide funding to the various teams. I and Alana, we are part of the Health Science Futures Office, which is much more forward thinking. It's trying to take those science fiction-like ideas and proving that they're possible. The Scalable Solutions Office, they are focused on ensuring that everybody can have access to those healthcare solutions. So scaling up and scaling out, reducing cost, manufacturing, so that geography and money are not barriers. The Proactive Health Office is primarily focused on preventative healthcare, ensuring that we can develop tools and technologies that enable us to be healthy longer throughout our lives. The Resilient Systems Office is more of a systems level office, focusing on the healthcare system, whether that's cybersecurity, whether that's broad hospital systems, and so on and so forth, and to ensure that the healthcare infrastructure is functional in times of distress, or so that it's not brittle during, for example, like another COVID outbreak. And lastly, the Office of Commercialization is an office that helps to transition out the developments, the tech that we fund in some of the other offices. They help with regulatory insights, with commercialization, with VC partnerships, all those things that are needed to transition beyond that valley of death, so that what you develop doesn't sit on a shelf and gather dust. A little bit of detail on Health Science Futures, so you're kind of aware of what we're doing here. We really focus on the engineering discoveries, utilizing some of the more hard sciences, physics, chemistry, engineering, to but applied to biology. That's where the AIR program fits into our Health Science Futures portfolio. We are really kind of focused on in silico biology, so using some of the machine learning AI tools that have come to the fore in the recent years to simulate and model biology so that we can do things in an in silico system that we couldn't do otherwise in a laboratory, and therefore advance the science and the biology that way. We are looking to reinvent medicine so that it's curative rather than treatment of diseases and disorders. And ultimately, we also are involved with regenerating healthy cells, tissues, organs, so that those disease states can be replaced and regenerated. Some of those programs fit into those areas. The NITRO program, the PRINT program, or others kind of fit into that regenerative medicine space. Alana's other program, Psi, on surgical precision and visualization, kind of spans a lot of those things. It takes elements from physics and engineering, but also some computational tools and regenerative medicine to identify the cancer and excise it as needed. We have programs on transplanting an eye. We transplant livers, hearts, kidneys, but why not the eye? It's because it's extremely complicated to reconnect an optic nerve and produce that visual signal from the donor eye to the host's brain. We're looking into ways of facilitating that. This is just giving you an overview of the space in which we are thinking of the future of health and hoping to really kind of generate that inflection point, that threshold for an action potential that we can solve the health care problems of the future. With that, I think I'll turn the time over to Alana.
What if we could perform life-saving interventions autonomously? So that's sort of the high-level view for this program. What are we trying to solve? And essentially, we looked around and we realized that where you live sometimes determines if you live. There are many, many areas in the country where if you happen to live there or just be there, if you have a stroke, your chances of making it out without disability are extremely low. And that's something that's not okay because stroke is very treatable. If you are very close to an emergency room, then you're home free. You don't even have to have disability, right? So that's sort of where we're going to. I gave a little bit of a history. So we were disease and modality agnostic when we started to look in the surgical field what's out there. So we received information through the RFI, and then we organized a workshop in 6 broad different areas. So we looked at endovascular, endoluminal, laparoscopic surgery, ortho, ophthalmic surgery, and microbots, right? So there were separate groups, and we tried to soak in where the field is, what's needed. And essentially where we want to fit in is catalyze developments that would not happen without us providing an infusion of capital. And we felt that the 2 areas that would actually make the most sense to catalyze would be endovascular robotic system and microbots. The other 2, like for example, in orthopedic surgery or in ophthalmic surgery, there are already companies that are relatively close. Laparoscopic is sort of a very broad area, and it's really hard to do one thing that will be impactful. But what struck us is that in particular in stroke, there is a very clear way to get there, right? So technically it's really hard, but it's not impossible. I think one of the issues that we realize is that technically speaking, it can be done. I think it's more the regulatory aspect, and it's also the LC implication, ethical, legal, and societal implications, right? So would you be comfortable being operated on by a robot? So not only that we have to put this program out and ask you to make it happen technically, but we also partner with the FDA. Of course, nothing like this has ever made its way through the FDA for them to understand what do they even need to require to make sure that the system that is proposed is actually safe, right? So part of this program will be the partnership with the FDA, and Aldo will give more of an overview of how they will work very tightly with the performers for technical area 1 to define what the requirements and what the test should be to ensure that the endovascular robot is safe. Right, so in a sense, if you wish, we feel that technical area 1 is sort of TRL level 3 or 4, technical readiness level. The other one that we chose to address are microbots. And that's a little bit of a crazy offshoot there. But we also realize that there are entities out there who do this work, and it's grunt work, and it's really not supported. And we felt that in this area too, we can make a difference. So that's how we narrowed those 6 areas down to 2, because we felt that in these 2, we can make the most impact. So the program vision is that we would develop autonomous systems that perform surgical interventions. There are 2 areas. One is technical area 1, which is endovascular robotics. These are essentially procedures delivered through the blood vessels. And obviously, if you had been in the field, you know that the most important thing that needs to be done is clot removal after stroke, right? So that's what will give you the best bang for the buck. But we don't want a system that just does one thing. So we're also asking performers to demonstrate embolization and to demonstrate angiography. Angiography is sort of normal. Clot removal. So if you do clot removal, you have to do angiography. Embolization is a little bit of a different beast, but once you have the system put together, you should be able to demonstrate embolization as well. So we're looking for end-to-end solutions, right? We're not looking for a fancy catheter. We're not looking for, a fancy way to extract the clot. And we fully understand that in your labs, you actually may only do one of those. But as an ARPA, our end solution is not to develop science, not develop fundamental understanding of science. Our fundamental mission is to develop solutions that save lives, right? So if you just develop a catheter, great, amazing, but we're not interested, right? So bear in mind for this program that we are looking essentially from point A to the end. We understand it's going to take more than 5 years. I'm sure that by the time I leave ARPA-H, there won't be a robot crawling through my veins. But we want to see the red line between where you are and where that's going to make an impact on people's lives. So it's a good opportunity here if you're only working on a part of that. For example, say your lab or your company is working on autonomous navigation, great. Make sure you have the whole chain spelled out in one proposal. Technical area 2 are microbots, and there are slides detailing what we mean by microbots because many people mean different ways by microbots. But essentially, we're asking you to develop small entities that can move, sense, and act. And I will underline the word act. We don't just think sniffing around. We want things that do something, okay? So the act part is really important. What they do, in this case, may be a little different than the typical ARPA-H project, where we're sort of specific in terms of what are the metrics and milestones. For the second technical area, we are not. We give you a lot of freedom in basically spelling out what the microbot is, what it does, and how well it should do it, right? So for TA1, you will see very clean and very crisp metrics and milestones. For TA2, because we allow such a broad range of options, you will have to spell out your metrics and milestones, and we will go back and forth in the negotiation process to make sure that you're going fast enough in that area. So this is more of a, again, TRL 2 or 3. It's a completely new treatment modality. It can be adaptable to many interventions, and essentially we want to catalyze you thinking. And traditionally, we have the proposals due 2 weeks after Proposers Day. So we should have asked for the proposals in January, but we realize that in particular this area, it's usually something that you won't have sitting on the shelf. So we delayed the solution summary submission until later in January because we wanted to make sure that you have time to think about what you propose. So endovascular surgery, what are the current limitations? And I'm very grateful to Kurt Yeager. So he is an endovascular neurosurgeon, and he was kind enough to put up with us for a few days and let us observe what he does on a day-to-day basis. And obviously, embolizations or angiography is their plan, but we actually insisted we wanted to see how a stroke is treated. And we booked a hotel right next to the hospital, and he pinged us at, 12 or 1 a.m. when the patient arrived. We picked up and we ran, and we actually observed how it's done. So I think it's really important you read the literature, but essentially you find anything in the literature. So we felt it was very important for us to have a full understanding of what the current situation is and what the limitations are. So number one, there is a severe surgeon shortage. There are only about 1,100 endovascular neurosurgeons in the US. It takes a 7-year residency and a 2-year fellowship. So really, there aren't enough people to do these life-saving procedures. That's why a robot should come in. The tools are also, I'm a physicist by training, sort of an engineer, biomedical engineer, but it was mind-boggling on how you basically look at the angiography, you figure out, well, maybe I should use this catheter because I need to take this turn, and you try to take the turn, and it's like, well, that wasn't the right catheter, so you have to yank it out and go for another one, which really slows you down. Time is brain. That's not an ideal situation, right? So the tools that they have are not great, and they kill time and they kill brain. Also, the surgeon looks at the 3D angiography, but then during the procedure, the best you have is a biplane angiography. So the surgeon has to do the registration in his brain, figure out where he's going. Now, in this day and age, with registration and AI all over the place, like that's 3 centuries ago. And the other thing that is interesting is you get a lot of harmful radiation as a patient. Assuming you make it out, it's not ideal, but it's also bad from the surgeon's perspective. We only have 1,100 of them. And there are a lot of cases of reported left eye glaucoma in the surgeons because they're always exposed to radiation from the left-hand side. And that's something that ideally we would like to take care of because it is an occupational hazard for the neurosurgeons. Okay, so I was arguing that stroke treatment is a critical unmet need. And if you look at the statistics, they're flabbergasting. So there are more than 900,000 strokes each year in the United States. And out of them, about 775,000 are ischemic strokes, those that are caused by a clot. If the obstruction is in a small vessel, then TPA will do the job. But if the obstruction is in a large vessel occlusion, then TPA is not enough. And the standard of care is to perform a thrombectomy. And we have on the order of 335,000 such strokes that happen in one of the large vessels. Depending on when they get to the hospital, about 160,000 of them are called salvageable tissue. And that's a little bit of an unusual situation because the thought process was that you should only do it within 6 hours. But now, after clinical trials, the understanding is that it's actually beneficial to the patient even up to 24 hours. So in fact, there are more than 100,000 people who would be candidates for a thrombectomy. And yet, at the end of the day, there are only about 40,000 patients who get this thrombectomy. So that's almost 300,000 people that are not treated. And you wonder why. And what you see there is a map of the United States and where the thrombectomy-capable centers exist. There's only about 300 of them in the entire country, and about 50% of the Americans live more than 1 hour drive, and about 37% of them live more than 1 hour flight for a thrombectomy-capable center. So honestly, if you live in the middle of the United States, just don't have a stroke. Obviously, time is brain, and every 10 minutes of earlier treatment results in an average gain of about 39 days of life without disability, and it saves the healthcare system about $10,000. So really, the faster you treat, the better off the outcome is for the patient. And last but not least, stroke is the number one cause of disability in the United States. We spend to the tune of $56 billion a year just for stroke treatment. So really, thrombectomy is a huge issue, and a lot of people in this country do not have access to it. So that's the main reason for which we had focused on thrombectomy as one of the AIR main goals. Now, switching gears, microbots. And we foresee this as a new modality for surgery. You look back in the past at where we started and where we all started at open surgery. Now, where we evolve, a lot of the procedures are done in a minimally invasive way through laparoscopic procedures. And going forward, why not do some of these surgeries with untethered microbots? Now, obviously, it's very clear even up front that not all surgeries will be amenable to microbots. If you have a 2-pound tumor, lo and behold, it likely will not be a microbot performing this surgery. But right now, surgery remains dangerous. It has the greatest risk of adverse events of all medical procedures, and there are complications in 8 to 20% of the cases. Also, very importantly, it matters where you live. So if you live close to New York or DC, then you likely will get a skilled surgeon and your outcome will be better. But if you live in a place where surgeons do not get as many cases, then your outcome is very much likely to be worse. Why microbots? Well, again, there is a possibility that they can treat anyone anywhere. It should be a less invasive administration, less specialized training, precision targeting, as well as they can access certain deep-seated anatomical pathologies. So going back sort of to business, the program has 2 technical areas. So technical area 1 is autonomous endovascular surgery, and that's broken down into 2 sub-technical areas. One is the development of the autonomous robotic system, and the second one is a simulation platform. So we are asking you, the potential performers, to apply to one of these. So either TA1A, which is developing an end-to-end robotic system, or the TA1B, which is developing of a simulation platform. On top of this, we will be funding FDA CDRH, and their purpose in the program is to develop validation metrics and phantoms for the TA1 performers. So those working on TA1A or B will actually work very close to each other and also very close to TA1B. Because in the end, what you target, the capabilities of the system and the metrics and the performance really has to be tied to what the FDA team will decide is the end goal and what would render a system safe. It's really important, and Gina will have an entire slide. We are very keen on not having anybody apply to TA1A and TA1B at the proposal stage. You can do it, and Gina will go through a whole slide. We had a lot of questions as to how many proposals you can be part of. Gina will go through the details. At the abstract stage, you may apply to both, one as prime, 1 or 2 as sub. But if you are selected for a full proposal, we do not want you to be part of both. The simulation platform, our intent is to catalyze this entire system and have that simulation. So all the robots should be able to be replicated in that simulation software. The simulation software will become public at the end of the program, together with all the data sets, including angiography, fluoroscopy, and whatnot, which are needed for validating the performance of the robotic system. So TA1A and B are meant to be disjoint, because if one team member is part of both, then it will inherently favor that robot, right? So the two have to be disjoint. The technical area 2 is autonomous surgical microbots. These are defined as untethered. They can have some internal propulsion as well as some external propulsion. They need, again, we're looking for an end-to-end solution, not a cute entity that does a cute thing, but we have no idea how to put it there and how to get it out, right? So end-to-end solution, how you put it in, how it gets to the site of the interest, how it takes care of the business, how we get it out, or if you make it biodegradable, that's fine too. But end-to-end solution for what the microbot does. And we will get through that slide of how many proposals you can get to. Gina will take care of that. So a few more details about the technical area 1, the autonomous endovascular procedure system. So what would the teams have to develop? So first of all, you need robotic-ready devices. So you need to devise catheters that would be adequate for autonomous workflows. We also ask you to validate that system in 3 different procedures. So fluoroscopy, thrombectomy, and embolization. So that system that you have has to be capable of performing all these 3 procedures, right? So that's the catheter. The second part is planning the mission. You're likely going to start with a 3-dimensional CT angiography. The beauty of how ARPA-H does things is we're telling you we're telling you what we want the end goal to be, but we're not telling you how to get there. Right, so we assume that you're gonna navigate based on fluoroscopy, but that doesn't have to be the case. It's important to note that as part of our metrics, we ask for significant dose reduction. So you can choose to still navigate on fluoroscopy, but you need to plan the mission and you need to get there, but it doesn't have to be fluoroscopy. Right, so plan the mission. I am in the femoral artery or radial artery. This is my clot. How do I get from point A to point B? That's something that you, the performer, need to figure out. You need to navigate to the site, generate the intraoperative roadmaps in some way or another, as well as navigate the catheter to the clot site. You need to perform the therapy, which is make sure that you have contact with the clot, as well as activate the therapeutic mechanism. How do you get the clot out? And the last step that you're going to have to do is confirm and close, right? So retract the catheter, confirm that flow has been reestablished, and close the patient. It's important that autonomous way to insert the catheters are not part of the program. Right, so we assume that the patient will go to the nearest OR, and there will be some form of nurse or clinician or surgeon who will insert the catheter. So opening and closing, we will not fund them. However, once you open the patient, from that point on to the closing, it all has to be autonomous. Navigation, performing the procedure, withdrawing, all should be done independently. So just to put everything in perspective, the way we see, there are sort of 3 categories of technologies that have to get into this program. Number 1, navigation. Number 2, manipulation. And number 3, catheters and guide wires. And we have columns on where we are in the clinic today, what exists in the lab, and where do we see us after the completion of the program. So obviously navigation is pretty much manual right now. The surgeon will have to do this 2D to 3 registration in his head. There are some papers out there claiming that they can do autonomous navigation. However, if you look at the review paper, the conclusion is there are no reference standards, bias and poor generalizability across the studies, low level of evidence for clinical translation. So really, we can't do it right now. At the completion of the program, we fully see autonomous navigation, using preoperative imaging and some form of real-time sensing. In terms of manipulation, there is manual pushing and pulling and twisting. That's where we are right now. There are some companies who have roller cartridges for basic teleoperation, some external magnets that can be very large. However, where we see this at the end of this program is essentially autonomous and remote controllable systems with precise manipulation. So again, our goal is that at the end of the day, everything will be done autonomously. But we fully understand that there will be evolutionary steps, right? So you develop the system, we ask you to finish demonstrating this in cadavers. Practically speaking, the way this will be implemented is there will be a clinical trial with a surgeon in the room, such as the surgeon can take over if something goes wrong. So that's step number 1. Step number 2 will go to teleoperation. And step number 3, we will go to full autonomy. So we envision the end goal the same way we had self-navigating, self-driving cars. There wasn't NVIDIA had the self-driving car about 25 years ago, right? And they went to the National Transportation Board and they go, here, we have a car that works. And they go, great, how does it work? And they go, we don't know, it works. It's like, yeah, that's not how it works. And it took 20 years for that explainability to be built into the system, right? So we're aiming for full autonomy, and that's what we want you to plan for, but we fully understand that there will be this evolutionary step in the process to getting to full autonomy. And number 3 is catheters and guide wires. So right now the surgeon has a bunch of them. They all cost a non-negligible amounts of money. They look at the CT angiography and they figure out sort of what should work for that patient. In reality, they don't all work, and then you throw away $3,000 and you start all over again. We give you the choice of using the catheters and guide wires that exist out there right now, as long as the robot will be able to spell out very clearly, I need this exchange, and the nurse should be able to exchange that with what they have on the shelf. The other option we give you is you want to develop something new, that's fine, but that thing that you develop shouldn't really require exchanges anymore. So we really want to streamline the process, such as you only use one guide wire, one catheter to do the entire job. Right? So that thing should be steerable, and it should be able to do the business end without requiring exchanges. Right? So if we're going to pay for something better, it should be really better, such as we don't do exchanges and waste time in the process. So at the end of the day, we hope that at least some of you will choose to develop these steerable catheters that will be able to perform the entire procedure without exchanges. Timeline and milestone, and that's a very, very quick summary. I advise you to read the ISO when you submit, because this is a very superficial summary of the metrics and milestones that we have in there. It took a long time to generate them, and it's sort of a middle road between what we felt is doable and what the clinicians needed. We have milestones on all kinds of different dimensions, including platform development, imaging and system integration, autonomous navigation, therapy delivery, as well as performance metrics. How fast should you do it? There are also advanced development. Again, depending on what path you take, some of the metrics and milestones would not apply to you. So if you choose to do navigation that's not based on fluoroscopy, then those metrics and milestones do not apply. But there will be others that will be specific to your system. And again, bear in mind that this is a superficial review of what there is in the ISO. Look carefully at the ISO. Now, this was technical area 1, and honestly, when we put the program together and we pitched it internally, this didn't exist, right? We said we just want, one set of teams to make it work. And through the internal conversation, the idea is, you're going to have a few teams make it work, but you really do not have a common denominator. And then you're done and you wish them good luck, and then what? right? So at that point, we took a step back and we said, well, we really have to create a common denominator. We want to create a validation platform such as every single company or entity who develops robots is able to baseline themselves against the simulation platform, which should accommodate all the robots, right? So the selected performer, and it's very likely that in this area there will be just one. For the others, it's very likely that there will be multiples. We're asking you to develop the in silico testing and simulation software, right? So you have to take in the 3D angiography, you have to generate the fluoroscopy, and you have to take in the robotic system model, and you have to predict how the robot will do in all kinds of anatomies and navigating all kinds of geometries, right? It's really important for you to understand that we're asking you to make this public, okay? So if you're a private company who wants proprietary software, this isn't for you. The point of this technical area is to baseline all current and future endovascular robotic companies on one platform. So that's why the software developed here has to become public. The other aspect that we're doing is, again, we're collaborating with the FDA, and Aldo will talk a lot more about what they're doing. They will tackle a few aspects of the program. Number one is we don't want different companies to baseline their system on different geometries, and then we need to compare them and down select, and we can't do that if they're not doing the same thing, right? So FDA will develop phantoms, and will likely put out drawings of different heart anatomies and brain anatomies and CAD files for you to print these phantoms, such as everybody navigates, sort of the same phantom anatomy. This is a simplified, this is where you start, simplified phantom anatomies that will progressively become harder and harder. They will also look at evaluation metrics for autonomous interventions, look at safety, effectiveness, also give you simulation parameters. So no matter how thorough you are, you can only test it physically in, maybe tens of cases. But the reality is that the human anatomy is significantly more complex. And the third thing that FDA will do is develop a framework for validating this autonomous system. We don't want to let you go at the end of the 5 years where you met our metrics and then you go to the FDA and nobody has any idea on what's required for this system. So this understanding will be developed by the FDA in conjunction with the TA1A and TA1B performers. So be prepared to make quick friends with those people because you will meet them weekly. The other thing I wanted to mention, and it's not here, we actually commissioned an ELSI study, right? Ethical, legal, and societal implications for this. Even if you make the best system, it's not guaranteed to be accepted in the clinic, right? And we will pay for that in parallel for people who have experience in this to sort of warn us ahead of time what the pitfalls can be and how we can potentially address them by doing education or outreach or considering things that maybe we weren't going to consider upfront, right? So this will be done in parallel with the performers, but the results of the study will be made available to the entire community. And hopefully that will help speed up the transition towards product. Again, TA1B timeline and milestones, you see them there. The one thing that's important is that we are asking for the performers to acquire 2,000 anatomical CTs and MRI angiography data sets, as well as at least 500 fluoroscopy video data sets of endovascular procedures. So in order for you to navigate, you need to learn how to navigate. And those data sets do not exist anywhere right now. So the TA1 performer will need to figure out how they get them, and they will become public. So from that point on, anybody who wants to develop this kind of navigation will at least have the data. Switching gears, what's a microbot? And this is what we define to be a microbot. Number one, it has to be untethered. So if you, for example, develop a microbot that's meant for thrombectomy and it's tethered, you should apply to TA1A. If it's untethered, you may want to apply to TA1B. We're not going to throw applications out because they're the wrong place. Like, we are a small shop and we really treat you individually. So in that case, it's not going to be a big issue. The entities that are between 200 nanometers and 2 centimeters, we are considering some type of GI robots. Those, the gastrointestinal ones, can be up to 1 centimeter diameter and length up to 3 centimeters. The microbots can be one entity or it can be multiple entities. And again, they can move, sense, and act. I'm underlying act. We don't just want sensing microbots. We want microbots that do something. What do the microbots do? We're giving you some examples, but we're not limiting you. So, for example, they can biopsy, they can deliver energy, and that can mean stimulation or it can mean ablation. It can deliver cargo, such as drugs, cells, materials, staples. They can destruct tissue. They can do it chemically, mechanically, electrically, they can burn. They can do active monitoring followed by treatment. For example, surveillance, identification, and elimination of circulating tumor cells. Again, this is not limiting. These are the examples we give, but you can choose something else. What's out of scope, this isn't a drug delivery program. So we are not looking for biological solution, chemical solution, or a combination of the two, right? So nanoparticles or chemical sensing are not part of the program, and developments that tend to be pharmaceutical in nature, they're not part of this program. So please do not apply. A few more specs for microbots, what's acceptable and what's not acceptable. So composition, they can be mechanical or electronic, or they can be hybrid. What does it mean? It includes at least 2 components, and at least one of which is either mechanic or electronic, right? So it can be a cell that has a circuit on top of it. That works. The components must be either biocompatible or they can be properly encapsulated, right? So please do understand this is not a traditional drug delivery program. What's not acceptable, purely biological, not okay, purely chemical, not okay, chem-bio hybrid, not okay, nanoparticle, not okay. Okay? How do they propulse themselves? They can have internal propulsion, they can store energy in some way, and please make sure that if you're applying in this area, make sure that you spell out that you can have enough internal energy to do the job. The propulsion can also be external, untethered, but it needs to be card size. We are not looking for magnets the size of a room. So we are not looking for untethered large external motion like large robots. We're not looking for MRI systems, and we're not looking for physical tethering to something on the outside. Power generation can be both internal and external. We don't want anything that's larger than a cart. And in terms of the GI microbots, we are accepting ingestible GI microbots that can sense and biopsy or sense, biopsy, and ablate. We are not accepting ingestible imaging microbots or microbots that deliver chemical or biological agents. Again, we are not limiting you, but we're giving some examples of what we think. We read all the literature out there. These are examples of the things we think you can propose. So, for example, in the cardiovascular system, you can have cell-tethered electronics for targeted therapy. You can have circulating tumor cell identification and killing. You can have thrombolysis. GI system, identification of polyps, biopsy and ablation, or mechanical or thermal lesion closure. In the urinary system, you can have disintegration of bladder or kidney stones or targeted bladder cancer treatment. In the reproductive system, you can aid in vitro fertilization, intrafallopian transfer. In the respiratory system, you can have targeted infection, or you can have treatment of lung nodules. In the nervous system, you can have intrathecal drug delivery. or CNS milieu modulation. These are just some examples. Do not feel limited by those. You can propose anything else, ensuring that you are able to spell out an end-to-end solution. The timelines and milestones, again, are very different than TA1. So with TA1, we were over-specific, or TA2, we couldn't, because there is a huge range of what you can propose. So you will have to come up with the metrics and milestones. You have to be able to explain to us how they move, how they sense, how they act, and how you get them out at the meeting. Okay, so make sure that when you write the proposal, you spell your own metrics and milestones. And again, this is a synthesis. We give some examples of what we expect you to do if you choose a microbot in a certain class in the ISO. This is an example. For example, if you have a cancer targeting immune biohybrid, that's what you expect you to do. So by motility, more than 80% of the injected cells get where they need to go. You need to be able to confirm that they got where they needed to go. You need to be able to confirm their action and how you get them out. Again, this is just an example. You have to come up with your own specific write-ups. And timeline. I have good news and bad news. I'll start with the good news. And the good news is that we're really working hard to put this under contract by the end of June, which honestly is insane. But we're really working hard to get there. And that meant we have put the timelines initially. The solution open submission was on January 26th. We pulled everything down earlier, right? So your submission deadline for solution summaries is on January 20th. We will take, it depends how many solution summaries we get. I expect you should have the answer in no more than 2 weeks, whether we encourage or discourage you to submit a full proposal. I want to be very clear. We're not telling you yes and no. You can choose to get a discourage and still submit. But when we say discourage, we mean it. We keep at least 3 times as many submissions as we know we can fund. So the fact that you got a discourage means you're very much far away from where everybody else is. So if you choose to spend 6 weeks full time to write this, by any means, have at it. But do understand that discourage is not taken lightly. So if there are aspects of the submission that we think can be good, we'll give you an encourage. Your full proposal will be done by March 18th. And just bear in mind that this aggressive timeline will mean that your contracting officers and your administrative officials and everything will have to move at breakneck pace. You and us. But the hope is that everything will be under contract by mid-year. And that's all I have. And I'm 1 minute early. Thank you. And I will let my colleague, Sen Gassman, talk about what the Office of Commercialization is and how they can help performers transform their developments into products.
At ARPA-H, the success of our programs isn't measured by the number of publications they generate. We measure success by the technologies that are created and the impact they have on the health of patients. And so while the mission of ARPA-H is to develop moonshot technologies like microbots, we also can't fund them in perpetuity. At some point, they're going to have to graduate and stand on their own. My name is Sam Gussman-Toe, and I'm speaking on behalf of ARPA-H's Office of Commercialization. So our job is to make sure that ARPA-H technologies stick the landing. We're partners for our performers, and our goal is to build your capacity to succeed after funding is over. So if you're selected for the AIR program and become a performer, we have a variety of support options that are available. These include regulatory consultants, IP experts, reimbursement experts as well. We can help with transition road mapping, so helping you plan out the different activities you're going to need to do and when you're going to need to do them. We even have entrepreneurs in residence who are experts from industry, who have successful track records, who can work with you to help you develop your commercialization strategy and help make sure that you have what you need to succeed long term. But I also want to say that for now, for this presentation, I want to focus a little bit more on transition and commercialization and what types of challenges you might face. So that way you can think about it as you're putting together your proposals, but also your teams, so that you can try to build complementary teams that are going to be able to take your technologies that you're building and make sure that they actually make it to the market and make it to patients. So transition has many dimensions, and unfortunately, good R&D isn't enough to ensure success. You need to make sure that you're addressing the holistic elements that can impact your ability to develop a product. And so while there are many different commercialization frameworks out there, and I'm not going to claim that one's any better than the others, what I have on the slide is one that we use to make sure that we're checking the major boxes. And I think it really highlights a lot of the key activities that you need to perform, but also it raises some important questions. You're going to have to start by building a product around your technology. And this means you really have to understand your users and the workflows that this is going to need to integrate into. But at the same time, your users aren't necessarily your buyers, right? Often they're very different. So who are the buyers that are actually going to need to decide whether or not to purchase this technology? And what are the different economic value propositions that are going to drive them? And what's the different data you're going to need to generate to show that there is going to be a return on investment for shelling out for these amazing new technologies? And while you're focused on the business model, you also have to be thinking about your IP. How are you going to manage your IP and protect it? And are you going to make sure that you've licensed all the underlying IP and there's nobody out there that's in a potential blocking position? Regulatory is another significant consideration. Do you know what pathways you're going to go through and what evidence you're going to need to generate? And if you're doing something unprecedented, how are you going to be engaging with the FDA early? And we're very lucky here with AIR to have them involved. But how are you going to be handling that, and what's your plan? And probably most importantly is thinking about what is the end game. What's your exit plan? One option is to build a empire around your technology, right? But that might not be realistic for everybody. Maybe what you really want to do is to license your technology out. So those are some general considerations from my spiel, but I also want to try to give some actionable advice too. Hopefully you'll find this helpful as you start working on your proposal. So if you develop a full proposal, you have to create a one-page commercialization plan. And so we know that this is quite short. Like realistically, you could write a dissertation about it. But so that means you're going to really need to prioritize the most important elements for your commercialization plan. You're going to need to make sure that you're highlighting the biggest challenges you face and that you're demonstrating that you really understand what's entailed in bringing your technology to market. Also, I want to just mention, it's really a good thing to acknowledge important unknowns that are out there. We don't expect you at this stage to have all the answers in all of these areas, right? But showing that you understand the problem and what it's going to take to bring your technologies to market is really key. So when it comes to your transition partners, what is your plan? Are you going to have a built-in transition partner on your team? So is one of the organizations in your team going to carry your technology forward after it's been developed? Or are you planning to spin out a new startup? Or, as I mentioned earlier, are you going to try to license your technology? The next area you're supposed to address are important bottlenecks and hurdles. So, what are going to be the top challenges that you need to face to bring your technology to market? Is it going to be on the regulatory or reimbursement side? Maybe it's dealing with clinician and patient adoption. Or liability could also be a significant factor and challenge that you need to overcome. Market access is always a consideration. So do you understand the market, its size, and who the buyers are within it? Do you understand the different reimbursement pathways you'd be following and whether you can fit into existing CPT codes or will you need a new one? And what's the different evidence you would need to generate for coverage determinations? And how can you build that into your clinical trials and studies to make sure that you're generating it in parallel and don't have to do additional studies? For sustainability, what are the main revenue streams that you would be mapping out? What are the resources that are going to be required to operate and to execute? And then what's the different ongoing support that you're going to need to be providing to make sure that your devices can actually be used in practice? Is this going to be training, maintenance? What is it actually going to look like? What are the different services that it entails? And also IP too. How will you do your IP management? And so for the last element is scaling. And so, this is kind of defining your growth strategy. Do you understand who the early adopters would be, right? Who are you going to start targeting first? And what are the capital needs that you're actually going to need to be able to grow? And how quickly do you think that you could do this? I mean, you should really be thinking, how am I going to get our technologies into the hands of as many people as possible, right? It's a challenge. So I know I'm raising a lot of questions, and I don't have answers, and you won't too, right? But I'm really trying to just prompt you to start thinking about these. I have a few specific considerations I also want to share for each of the technical areas. So for TA1A, it's really a huge benefit that we have our partners from the FDA participating in AIR. But it's really not going to solve all your transition problems, and it's not going to make the regulatory elements go away either. You're still going to need to run clinical studies and generate evidence and make sure that you understand the pathways. So what are the pathways that you expect? Then there's also the reimbursement element. So do you understand what is your reimbursement strategy, and like what's the evidence that you're going to need to generate for it? And additionally, I think another element that's going to be really important for TA1A, for the autonomous systems, is liability. So how's it going to be handled if the system ends up making a mistake? It's something that planning ahead for can really help spur user adoption down the line. Another thing that is important to consider is, how are you going to make this affordable for small and rural hospitals? They're the ones who stand the most to benefit from it, but they don't necessarily have the capital budgets that major health systems do. I've spoken with hospital administrators who have, a $500,000 a year annual budget, right? For so many small rural hospitals, something like a financing option readily available will make or break their ability to access these kinds of technologies. So what's your strategy going to be for doing it? For TA1B, for the simulation, I know we're discussing open sourcing, and that's really great, but that's not a panacea. It's not going to solve transition for you, right? Just slapping an open source label on it and putting it on the internet doesn't mean it's actually going to be used or adopted or have a significant impact. So what's your strategy going to be for actually driving people to use it, for growing the user base? And then also, if you're open sourcing it, how are you going to maintain it and make sure that it stays current with technical developments? Are you going to need to develop an organizational support structure around it, or partner with a nonprofit maybe to do that, right? So what is the overall strategy going to be for how you address these elements? For TA2, for the microbots, for those of you out there, it's very nascent, but you have some amazing challenges ahead of you. One of the first and foremost things that comes to mind is, how are you going to be building trust with patients and clinicians, right? People aren't going to want to use something they don't trust and don't understand. And, regulatory, I believe, is also going to be have some uncertainty with it. How is this going to be regulated? And, what are the different kinds of studies you're going to need to do? Like, how are you going to prove the efficacy and safety of this? Starting to think about that ahead of time is really, really going to save you so much later down the line. And again, similar for the autonomous robotic systems in TA1A, how are you going to address liability, right, if something does end up happening? I think another important element for the microbots is understanding who the early adopters are going to be for it, and what are the different use cases that are so pressing it's really going to drive people to want to be early adopters of your technology, right? You're going to want to find things that have real value propositions that are going to spur people to be interested in the technologies. So I'm raising a lot of questions here, right? And I don't have the answers, and I'm hoping all of you, since you're way smarter than me, are going to be able to come up with them. But these are the kinds of things and the types of considerations that you're going to want to put into your proposal and into your commercialization plans. And also you're going to want to discuss with your teams too, as you're putting together teams to make sure that you're aligned on what the long-term visions are going to be. So I'm going to be around today if you have any questions. I'm more than happy to answer them in the breaks. I'm happy to talk about what the Office of Commercialization does and how we support performers. We really do offer a variety of support, and we'll really, if you're selected for the program, we'll really go in depth on that at the program kickoff. The main thing to remember is that if you ever run into any commercialization or transition challenges as a performer, you should bring it to Alana's attention proactively. She's a force of nature, and she's going to work with us to make sure that we get you the resources that you need. So this is the best part of my job, getting to meet performers and proposers like yourselves and hear about your ideas. So thank you so much for the opportunity to present today.
My name is Georgina Perry. I'm the Agreement Officer for the AIR program. And the purpose of this portion of the presentation is to provide you with the contractual requirements as you are preparing to draft your solution summary and to hopefully answer some contractual questions that you may have. The AIR program was solicited utilizing the Innovative Solutions Opening, also known as an ISO. The ISO was selected as the solicitation method for this program because an ISO allows for merit-based awards to be issued. It allows for more than one solution to be proposed to a problem, and it values technical quality over price. Under the AIR program, all the awards that will be issued will be other transactions. In other words, there will be no FAR-based procurement contracts issued, and there will also not be any financial assistance contracts issued. That includes grants and cooperative agreements. Only other transactions will be considered. We invite all non-government entities to participate if they believe that they have a solution for the AIR program. We intentionally do not provide a funding limit or a range for any individual award. And the reason why is because every solution is unique to what you are proposing. So your proposal should mirror the solution that you are presenting forward. Solution summaries are mandatory. Failure to submit a solution summary will prohibit you from being able to submit a full proposal. The government will only consider those solution summaries that have been submitted by the submission deadline and dates. Proposers who submit a solution summary may submit a full proposal regardless if you are encouraged or discouraged. But a caveat to that is that when a decision is made not to encourage you to submit a full proposal is because we recognize that there is time and money that is put into putting a full proposal in. So we do ask that if you do submit a proposal, if you are discouraged, realize that there is a risk with that. The government does not anticipate reviewing any non-conforming solution summaries or evaluating non-conforming full proposals. I think the important thing to take away from this is that all the information that you need in order to submit a solution summary or full proposal is in the ISO. So please do read the ISO in its entirety. As stated earlier, only other transactions are the types of awards that will be made under this program. Other transactions are a form of a contract. However, they're just not FAR-based contracts or procurement-type vehicles. The other transactions are designed to reflect commercial contracting practices rather than traditional federal procurement contracts or financial assistance vehicles. Other transactions is a method for us to reach non-traditional contractors that in the past may have not wanted to work with the government, but with other transactions, there's a little bit more flexibility and less guardrails versus a typical FAR-based requirement. With other transactions, there's an expectation that there'll be increased collaboration and partnership, leading to more effective use of resources and knowledge sharing. And one of the other benefits of other transactions is that you have an opportunity to negotiate the terms and conditions with ARPA-H, unlike FAR-based contracts where there is clauses and provisions that cannot be negotiated. Eligibility information is provided under Section 3.0 of the ISO. We encourage all participants that are interested in submitting a solution summary to adhere to the eligibility information that's provided within the ISO. But there is a caveat, though, that government entities, such as federally funded research centers, are not allowed to submit to the AIR ISO. We request that everybody have a SAM registration completed and active at the time that a full proposal is submitted. We do not require that the principal investigator have a SAM record, nor do we require that the subawardees have a SAM record as well. By statute, ARPA-H will prioritize awards to entities that will conduct the work in the United States. This, however, does not prohibit non-US entities from participating, but we do ask that if you are a non-US entity, that you partner with a US partner or entity in order to be considered for an award. Submission and selection process overview. The solution summary date has been moved. It is a mandatory submission. The submission is January 20th at 5 p.m. Eastern Time. The templates and the guidance for the solution summary are provided within Appendix A. Every solution summary that's submitted within the due date and time will be reviewed and a response will be provided. During the solution summary stage, you will be either encouraged or discouraged. For those proposers that do reach the full proposal stage, submissions are due May 18th at 5 p.m. And the instructions for the full proposal are under Appendix B. There is 4 volumes that you need to look at within the templates and submit in order for us to be able to consider your full proposal. And as Alana stated, we do have a lock date of June 30th to award all the selected proposals under the AIR program. So we ask that you pay close attention to the draft OT that's provided with the templates. We ask that you provide red lines to those terms and conditions, and that's an opportunity for you to let us know that you want to negotiate some of the terms and conditions. We also ask that you complete all the admin and national security policy documents, and this includes including a bio sketch, common forms, the support forms, and any information for key personnel, including the prime, sub, and sub-consultants. The full proposal will be evaluated against criteria 1, 2, 3. And some of the key highlights to remember is that each step included in the notification process will occur as quickly as possible because, once again, we're dealing with a shortened timeline and we plan to make all awards by June 30th. We request that you adhere to the full submission requirements and to make sure that you are reviewing the eligibility and conformance sheets before submitting that proposal. Evaluation criteria and descending order of importance. The first one is overall scientific and technical merit. And within this criteria, we're looking for innovative, feasible, achievable, and complete solutions. We're looking at the technical risk and the feasibility of the mitigation strategy that you have proposed. The second criterion is the proposer's capabilities and our related experience. We're looking for your overall team experience and expertise. We're looking to see if you have the capabilities to be able to carry out the solution proposed and to be able to manage the cost and the schedule. The last criterion is the assessment of proposed cost and price. The price analysis will be performed to assess the reasonableness of the value of the overall proposed price. And once again, since we have not provided you with a budget or a ceiling, we ask that you propose a budget that mirrors the solution that you're presenting forward. Applying to multiple TAs. Teams will not be awarded as a sub or a prime on both a TA 1A and TA 1B to avoid a conflict of interest. In other words, the software is meant to be independent of the robot and work equally well with all robots. A single team may not be the prime on more than one award. However, you are allowed to submit up to 3 solution summaries and/or 1 full proposal and 2 as a sub. I know this might be a little bit confusing and we may get some questions on it. But for the solution summary stage, we ask that the solution summary stage actually is a little bit more open because you're going to be able to finalize your decision if you want to work on TA 1A or TA 1B at the full proposal stage. So a team may not be part of both TA 1A and TA1B at the full proposal stage, even if they're only a subcontractor. And what is considered a team? This is a question that's been presented forward several times. Under university, a team is generally a lab or a collaborative group where there is a distinct PI for each group that is being proposed under that university. For a business, it's typically the legal entity that has a unique UEI, and that would equal one team. The two independent subsidiaries of the same parent company may apply and be awarded as a prime on a TA1A and TA2, or TA1B and TA2. Two independent subsidiaries of the same company may not be awarded on both TA1A and TA1B. Some final guidance. I think it's so important that the ISO be read completely before responding to the ISO. We just want to make sure that you understand that nothing stated during today's Proposers Day is contractually binding unless an amendment has been issued. And to date, we have only issued one amendment. The amendment revised the due date for the solution summary, it revised the due date for the full proposal, and provided clarified information on the teaming arrangements that ARPA-H will consider under the AIR program. We ask that if you do have questions, we ask that you first visit the program page, review the frequently asked questions, and attempt to obtain an answer through one of those postings that we may provide. But if you're not able to have your question answered through that, we ask that you email us directly at air@arpah.gov. The government does not intend to respond to inquiries via email unless it is a question that we have not been able to answer via the frequently asked page. Frequently asked questions will be posted to sam.gov and will also be posted to the AIR program page. We want to remind you to please register and update your UEI and account information in sam.gov. And we want to remind you that the ARPA-H Solutions Submission Portal is the portal that you'll be utilizing to submit both your solution summary and your full proposal. The last guidance would be to make sure that you are reading the ISO. I'll go ahead and turn it over to Jennifer Myers, who's the BFM.
A lot of what I'm going to discuss is going to be repeating what Gina has told you about reading the ISO, the requirements for the solution summary. It's 4 pages. We are going to ask for a rough order of magnitude cost proposal. If you're encouraged to submit a full proposal, then there are 4 volumes that are required. The technical volume that is going to offer your end-to-end solution to the AIR, I guess, problem, if you will. 25 pages is the limit for that volume. The cost proposal will be an Excel workbook, and any supporting documentation for the cost. And we really do want the PDF cost pricing backup for that. The national security information is going to be a requirement. And then Gina discussed the draft OT where you'll redline contractual information. All those will be required for the full proposal. This is just a screenshot of the rough order of magnitude, very high level, full costing, fully burdened costs. The cost proposal. This is where, as the business finance person, this is where I really enjoy going through stuff. So making sure that all the personnel costs are accounted for, really think out your supplies and timing of supplies. Will there be tariffs, right? Making sure that the prime proposer as well as any subrecipient proposer provides us a full proposal within an Excel workbook, submitting the vendor quotes, any backup that's going to help us validate the costs. Again, like Gina said, there's no funding limit on individual awards. We're not offering a range. We are going to be looking for reasonable proposals and that the technical team sees that the value correlates with the technical solutions. Super important deadlines. Your Q&A deadline is January 14th, 5 p.m. Eastern Standard Time. Solution summaries, January 20th. And then full proposals, if encouraged, March 18th at 5 p.m. I always like to provide a little cheat sheet of do's and don'ts. And you've heard it now several times, but it's the first thing on my do is read the ISO and really, really fully understand what we're looking for. Providing all of I'm focused on the cost, providing all of the cost proposal backup. That's really going to substantiate what you're trying to get across to us. Supporting documentation, including the subcontracting, subrecipient information for us. State how the costs proposed are realistic. It helps draw the line, especially for the finance people to get across to the technical people that we're capturing everything. The don'ts. It's a short list, but an important one. Please don't change the formulas within the Excel workbook. It tends to break the workbook. If you have to delete a row, just hide it, don't delete it. And, important, while we want teaming to occur, we do discourage cost strategizing. Please don't propose junior staff when really to get the technical work done, we need senior staff, just trying to get the proposal to fit or what you perceive our need will be. And with that, I would like to invite Aldo with the FDA to come up and talk about the FDA collaboration.
Thank you. Let me start by saying that I want to thank the AIR program team. It's been really a pleasure to work with Diana and everybody else in this collaborative component of the AIR program. My name is Aldo Badano. I'm in CDRH, Center for Devices and Radiological Health, FDA, and in the Office of Science and Engineering Labs. Our office is the mission of the office is to develop regulatory science tools. These are methodologies that innovators like yourself can use in early stages of device development as you gather the evidence towards an FDA submission. I have with me some other members of the team, and I will ask them to stand up so you can recognize them. Nirmal will be the program manager at the FDA side for the AIR program. Nirmal Soni is assistant director in the Division of Imaging Diagnostics and Software Liability. And Ravi Samala, who is here with us, is the program coordinator for the Artificial Intelligence Regulatory Science Research Program. There's also Ryan Beams over there, Sayed Kahaki, and Elim Thompson, as well as Andreu Badal there in the back. Please, I encourage all of you to talk to them, ask questions to us, ask questions. We'll stick around for another few hours to interact with you. The main team at the FDA also includes our office director, Ed Margerison, who's the executive champion for many of the things that we do with ARPA-H. I also want to say that while all the team that's here is from the Office of Science and Engineering Lab looking at regulatory science aspects of the program, we have started contacts with the other sections of the CDRH, which deal with the regulatory approvals or review. And our intention is to serve as a nexus between the proposers, the work on regulatory science, and the offices that will be in charge of looking at these devices when they come to us in a submission. And we'll do that through monthly updates with everybody. So I wanted to spend a little bit of time giving you a flavor of what is it that we do. So, some of you might be familiar with us, but I know some of you are not, and I want to make sure that you understand what our role is in this effort. Our mission is regulatory science, and what are regulatory science tools, you may be wondering. And as you can see in the screen, you have a range of documents that have different regulatory acceptance through the regulatory process, right? Of course, on the far right, standards are very well accepted. They apply to apply to a lot of devices, and sometimes it's just a checkbooks, right? You comply to a standard and good to go. If you go on the left side, then things are a little bit more involved. Guidance documents that help you submit evidence or submit your devices and guide you through that process. The medical device development tool program, which is fairly reasonably new, maybe about 10 years, that are tools that are qualified for a specific context of use. And then regulatory science tools, which are on the right of publication. So you publish your methodology, but then you make available a tool that's ready for this plug and play for your work as innovators. And the challenge there is that for some of the small companies particularly, we can publish a methodology, but then it will take you a few weeks maybe or a few months to read it, to implement it, to debug it, to validate it, and so forth. And by releasing these tools, we save that time for you. And I'll give you some examples of what those tools are. And that on the bottom left is the website. There is about 63 tools in the catalog at the moment, maybe 80. The first 63 are tools that we have some data on, and they have been using 1375 submissions across 536 product codes, affecting all 8 of our CDRH product offices. So these tools have wide range of applicability and are useful, and we continue to gather evidence to that effect. I'm showing this to position yourself in understanding our programs. We have in my division, we have 2 flagship research areas. One is artificial intelligence on the top, and the bottom one is what we call IRIS, which is interventional radiological imaging and visualization. And those are the priorities that we've identified this year, where regulatory science faces challenges in medical devices. And you see in AI, I don't have time to go through all of them, but data quality for AI training and testing, critical element. The emergence of multimodal models in devices, monitoring, post-market monitoring as an essential component where new tools are needed. Obviously, synthetic data playing a big role in the future, and then internal AI tools for our review. In the bottom line, IRIS, we have these 3 major efforts looking at bench testing, visualization aspects, and interventional. As you can see, there's great overlap between the IRIS program itself and with the combination of the AI into the AIR program objectives. So we're very happy to partner with ARPA-H in these areas. And again, I'm going to be very insisting in the sense that our involvement is on regulatory science. We want to work to deliver the methodologies that are needed to come up with the evidence that will be part of future submissions of products. And we want to do it with ARPA-H and with the performers. I think that we will all have to learn from each other in that process. Some of the key challenges, as we and ARPA-H have already identified, are the limited alternatives to clinical testing. Of course, you can do a large-scale clinical trial, which will take many years and probably a lot of money, and will be maybe applicable to your device, or maybe a version of your device. But clearly that's not sustainable, and bench testing has a significant role to play in any medical device regulatory review and approvals or pre-market authorizations. And on top of that, it was mentioned this morning, there's variability, right? You can test for some things, but there's a lot of variability, particularly in patients, and not only in the anatomy, but also in the lesions and also in the tasks. Because in this case, the AIR program has narrowed it down a little bit for us. So we are going to work on generating a testbed design with relevant properties for that purpose. We're also going to work on metrics. We spend a lot of time of our lives at FDA working on metrics. And in this case, it's a fairly new territory, as you know, in medical devices. Autonomy, we're seeing maybe in diagnostic imaging a little bit of that in terms of rule-out devices, which are devices that will autonomously look at an image and decide and make a decision on the presence or absence of a disease. That's really emerging, but that's only looking at an image. There's no intervention. So this is a challenge. And I think what we have determined is that to evaluate an algorithm of this type, where you're doing autonomous intervention, we will need a huge amount of data. You will need data for training, but then we will need independent data for testing. And there will be a synthetic data component to that. And I'm very happy to see TA1B, which is the generation of a platform that will be able to generate these images and data, not only images, but also data on the navigational aspects and the task completion and so forth. And then the last one is kind of to integrate all of that in a way that not only the performers will be able to use with ARPA-H, participating as well, but the community at large will be having access to those that framework so they can benchmark and compare the results. In the last few minutes, I'm not going to go through all of what I have here, but I will give you just a flavor of some of the tools that we've released in the last few years. And I'll start with the one in the left, which is a GPU-accelerated Monte Carlo X-ray imaging simulator. It actually was developed by Andreu Badal, who is right there standing. That's a very fast solution to in silico imaging that it has already been used in several applications, including breast imaging, CT, and fluoroscopy as well, and geography. So that's a tool that's ready to use and incorporate in the platforms. And let me just skip a little bit here, and I'm going to talk a little bit about this as again, just to give you an understanding of how what are our products that come from the Office of Science and Engineering Labs, which are tools in the area of synthetic data for imaging and AI. And I'll use this timeline just to give you that indication. What is it about? 7 years and 3 weeks, I think we published the first ever in silico trial, fully in the computer, that mirrored or replicated a clinical trial with patients. That was back in 2018. It was by today's standards, was probably a very simple in silico trial. But since then, we've continued developing tools that improved on that in silico pipeline. And here we have, we added the ability to do longitudinal studies, which means not only you have one lesion, but you can also grow the lesion in silico, so you can follow the performance across all different stages. We added a number of acquisition systems. Then later on, we released 10,000 images in a data set for mammography that developers of AI algorithms could use, which are very well annotated synthetic images. And we extended that to the pediatric case, which, as you know, is one of the areas where patient data is scarce and it's very difficult or complicated to get. And we've shown that if you train an AI for adults, which is what most people do, it fails when you see a pediatric case. And there's a reason for that, of course. It wasn't trained to see the type of objects that are in pediatric patients. And with AI supplementation, we can then achieve similar performance by without necessarily having to irradiate kids with X-rays, which is a plus. And finally, we went into the three-dimensional mammography. We just released a few weeks ago a data set of tomosynthesis images, which is fully annotated in terms of pixel level annotation. So that level of detail that the synthetic data provides, I think, might be something that we will certainly leverage in this program. You may get, and I don't remember the numbers for the data sets that are required for the AIR program, but you will have limitations in terms of if you bin that data into patient sizes or maybe other demographics, you end up with a number of cases. But to train these algorithms, you will need much more. There's no question. And then we will need to test it and test it over iterations. You'll have a version 1, test on a sample, and then generate new samples so that you can keep testing the new products that you have. I think I'm going to skip the rest of these slides, which show a tool where we evaluate the quality of synthetic data. And I'm going to maybe spend 1 minute on this one. One other tool, just to give you an indication here, is the MICMET tree, which is a decision tree for developers that are working on an algorithm for imaging and are wondering what is the exact metric or the preferred metric, I would say, that they would need to use when developing evidence for an FDA submission. And this is a tool that allows you to, depending on the type of device that you're working on, to get to the preferred metric. And with that, you also get additional tools that allow you to compute the statistical uncertainty around your metric and design your studies, which many of you maybe are familiar with some of these choices, but it's nice to have it in one tool, practical tool that you can use in a straightforward manner early on in the development of your device. Okay, and the last slide is this, which is kind of our initial thinking about the strategy that we'll have for interacting with the AIR program, really. Our interactions will be limited to regulatory science. Of course, we'll have our back channels to the regulatory offices as well, internally. And our thinking is that we will create a core regulatory science team that will be formed by some of our people from our office, some delegates from the performers, and then, of course, ARPA-H. And we will meet often to discuss the methodologies. We'll have proposals. I'm sure the performers will have proposals as well, and we'll work on it. Maybe we'll set up ways of validating our work and setting up test beds in different labs. We put together some initial meeting structure here, which might change, but it's going to be from our perspective, it's going to be a central effort, and we will convene that effort in kind of these ways, with having weekly, bi-monthly, and then quarterly updates that will be exposed to a broader audience at FDA. So I imagine that maybe every 4 months, the program will come together to FDA or somewhere, probably in this area, and showcase the progress in real science, so that we can have that discussion early rather than later to improve. So that's what I had. In summary, I want to emphasize that our role in this partnership is to develop open source bench test pipelines and data sets that will be available to the broader community through our RST catalog. And we hope you will, as performers, or those selected as performers, will help us to that goal. Some of the things that we'll deliver are the testbed platform, evaluation methods, pipelines, and datasets as well from our own in silico work. And those, of course, we always say that are voluntarily used in early stages of R&D, which is exactly, I think, what we're dealing with here, and then for later validation through the preparation of a pre-market submission. And I want to finish by emphasizing that the intent of this work in our office is to really make it easier for innovators so that by providing these methodologies, you can focus on what you do best, which is to generating amazing technology rather than on finding out how to evaluate it. So we hope that that's the realization. I think that's what I had. Yeah, thank you.
Thank you. All right, so we're going to go ahead and go into our Q&A session. Aldo's going to be part of the panel. Adrian Ruskin, he's one of our tech CETAs. Sam Gusman-Toe with commercialization. Alana. Paul Thienfrappa, he's one of our tech CETAs. Kurt Yeager, he's with Houston Methodist. And Gina Perry, our AO, as well as me.
So we will go as long as needed. Again, so please note, I know unfortunately my email is the worst kept secret, so it's out there. Please don't contact me directly because once the program is out, I'm not allowed to answer this question. None of us is allowed to answer these questions directly. The only way questions can be answered are here. So you ask them in a public forum and we answer them in a public forum. You can also ask questions online in the frequently asked questions page, and we will post the response. That will take longer. So the most effective way for you to get the answer is to ask it now. The majority of the questions we will answer now. There may be specific ones that we will have to delay the answer because I suspect you'll go, how about this and that technology, which will be in a gray area. And in some cases, we may decide to think about it before we give a final answer. So I will answer some of the questions and I will throw the others to my colleagues as well. So let's get the show started.
All right, my name is Andy Grant. I'm a vascular neurosurgeon at the University of Minnesota. So first of all, I just want to thank you for making this opportunity available. I've been dreaming about this for years, and I don't think that it would be possible without the effort that you put forward. So sincerely, thank you. The first question I had relates to embolization. So you had presented earlier that the proposed robotic solution needs to be able to do three things, one of which is embolization. The question I have is, can you tell us more about the unmet clinical need requiring embolization? And when you speak of embolization, are you talking about embolization of aneurysms with coils, intrasaccular flow diverters, embolization by placing pipeline stents, or embolization of AVMs, or embolization of MMA for subdural hematomas?
So I'll throw it back to Kurt for a little bit. So just he gives me his thoughts and then I'll give you my thoughts too.
I think this is meant to be a platform, or we want to kind of encourage a platform technology. So, embolization is part of that. So it eventually could replace a human doing these type of interventions. So I don't think we have any direct use cases, what you're referring to, but I think the goal is to kind of create something that can fill the void and do pretty much everything at some point. Maybe not starting with the most complicated cases, but eventually getting to that point in the future. But starting with a simple coil embolization or intrasaccular device placement, I think is probably the use cases maybe we would start with.
Because there I go back to the unmet clinical need. I think you highlighted the unmet clinical need for stroke, thrombectomy, remotely, autonomously to get out into the rural areas, and that time is essential. As relates to aneurysms, that time is not as critical. So is the unmet need that currently our therapies for aneurysms are not adequate?
So let me give you a little bit of context. When we first started it, we again, it takes 1.5 years to develop a program internally, and we have probably close to 200 stakeholders have a say. So the initial program actually only had thrombectomy, and the comment was, you really, as Kurt was saying, you want to develop a platform system. So obviously the highest impact is thrombectomy, but you don't want a system for thrombectomy. The fact of the matter is and remains that there's not that many centers that can perform endovascular procedures. So ideally, if you develop a system, you want it to do more than one thing. Right? So angiography, it's sort of natural. It's part of a thrombectomy anyways. But we wanted to highlight the fact that this system could do more. So if you want to choose one type of embolization, you can do that. We didn't specify it.
Okay.
But the thought behind requiring more than one is that ideally this maybe can be done in a hospital farther down the line that does not have an endovascular surgeon. So you can do an embolization in a local hospital. That was the thought behind it.
Okay. The second question is.
Can I quickly add?
Oh, go ahead.
We also encourage, we require that proposers think about what happens if there's a vessel dissection during the thrombectomy.
Sure.
And we encourage people to consider embolization as part of the solution. So like, even not as a separate procedure, this might be something that the robot needs to do as it's doing the thrombectomy. And that's really important to keep in mind.
That's a great point. The second is maybe a simple question is you had mentioned closure. And so just for a point of clarification, does the robot need to perform closure of the arteriotomy? Is that part of the solution? Is simple manual pressure an option?
No, the robot, so I apologize if we gave that intention, but it was very, the intention is clear. The opening and closing should not be done by a robot. So it can be done, it should be done by a human, by whatever means are commonly available. I mean, opening and closing an artery is not that hard to do. Any surgeon should be able to do it, right? So we want you to focus to everything in between opening and closing, but the opening and closing is not part of your problem.
Okay. The last question I have is around the issue of IP. If you have existing IP and you are selected for the development of a stroke robot and you use that IP, is there a risk that at the end of the day you lose that IP through this process?
No.
Okay. So if you're working...
again, maybe Gina can say a few words after I'm done. So these are other transactions, right? So if you go with grants or FAR-based contracts, you automatically get government purpose rights for whatever IP gets generated during the period of performance. I don't think anybody anywhere would reach back, right? So what you already have, it's yours. This is not FAR-based contracting, it's another transaction. So the type of IP negotiations we have can be less or more generous than government purpose rights. So in some cases, you can get more. Like, you know, there might be cases in which we may be willing to even give that up. And there will be cases, and we've negotiated that with a company for a year, we had to negotiate margin rights, because we are seriously concerned that that company would develop something and they would figure out they're cannibalizing their own market and choose not to productize it. And in that case, we had to, negotiate margin rights, saying if you're not making it a product within a year, within your constraints, obviously it's all a function of what FDA says, but if you're not taking these steps, the IP is the IP that you would have generated during the period of performance is ours. And then, or it's yours, you can license it, but we want to make sure that this technology evolves towards product. We don't want it to die anywhere. And I let Gina speak.
Yeah, no, Ilana is absolutely correct. You will retain the rights to that IP, that background IP that you bring into the project. The government's not interested in owning that IP. We are interested in government purpose rights, but you would retain ownership of that IP.
Okay, so if we were working with a company that had an IP for catheter technology and we were to apply that catheter technology to this project, there should not be a concern that they would lose that IP technology through this process.
Correct. There should not be a concern with that. So we just ask that we be provided with government purpose rights in order for us to be able to move to commercialization.
Okay, great. That is super helpful. Thank you again. I appreciate it.
Hello, my name's Jeff Berkeley. I'm CEO of Lightside Surgical. I was previously with Mimic Technologies, did a lot of modeling for a lot of the major robot companies out there. So my first one's just, I think, relatively simple. I know you're not allowed to be a prime and a subcontractor on the A and the B pathway or vice versa. What about advisors? Are you allowed to have advisors that can be on both?
So our main goal is to try and make sure that the software in TA1B doesn't favor anyone's robot over anyone else's robot, and that there's not even the perception of that favor in the case of like any sort of down select. So we do not want the same person being on a TA1A team and a TA1B team when it comes to the award. We are saying at the solution summary stage, right, like there's only going to be a relatively small amount of encourages. So feel free to be on multiple proposals, but once it gets to the full proposal stage, don't be on the TA1A and TA1B proposal, even as an advisor, because we don't want an advisor making it much easier for one robot to operate with the software than one other robot.
So will you facilitate, if we're interested in the B pathway, but you need robots and tools and CAD models and so forth to, build your system, are we going to have access to the robot team so we can get some of those models to test early as opposed to late?
Yeah, absolutely. Like the particular like cadence and the nature of this and the various like NDAs that I'm sure everyone is going to have to sign to infinity is going to vary depending on who gets the award. But like, yes, frequent collaboration will be encouraged.
And just to emphasize that we may actually consider the use of the software developed under TA1B as part of independent verification and validation for the team selected under TA1A. So it will be in their absolute best interest to dump those on you at T = 0.
Great. And final question regards the data sets. I know there's supposed to acquire a bunch of data sets. Clearly you want some real data sets, but are the numbers you're looking for synthetic data sets or are all those sets supposed to be real data sets?
Those that I have referenced, which is the 2000 CT angiographies and 500 fluoroscopies, the angiographies, the CT angiographies exist. You just go figure out where to get them from. The other ones, you will need to figure out partnerships to have them acquired because honestly, I don't believe they exist. So maybe Kurt won't, yeah. Yeah, we don't think they exist. So you are going to have to figure out a partner to go ahead and acquire them. So those are real. I mean, although we'll be doing some work from the FDA side for generating synthetic data sets, but the ones that we ask for are real.
And for the real data sets, will there be any pooling of the real data sets or is everybody supposed to come up with their own sets?
So we placed it in our responsibility to provide those. And the view is that those that you are acquiring through the period of performance will become public. Now, if you figure out somebody's giving you those by any means, I mean, obviously the performers will need those as well. If they have some and you want to negotiate with them to make those as part of the ones that will become public, it's up to you. We're not going to interfere. But the requirement is that at the end of the day, the ones that we had required will become public together with the software.
Okay, thank you very much.
Thank you. My name is Juan Vax from Purdue University. I have a specific question to you, Jennifer, regarding you said there is 20% a cost share that it's encouraged. So, and that's part of the do's list. I wonder, first of all, this is, for some institutions, this is very problematic, the issue of cost share. So I wonder at what extent that is, if an institution cannot provide that 20%, that encouragement that you mentioned there is something that may be used to discourage a proposal.
I would say that we strongly encourage partnering to be able to hit the 20% cost share. I won't say that it's required, but it definitely is encouraged.
So again, this mainly applies to TA1, not to TA2. And the main reason we want this is that we're really, really intent in driving the developments towards a product. And in order for that to happen, it will make the developments much easier if you have a company in some form partner at T equals 0. It doesn't mean that you can develop, spin out a new co, but it's much easier if you already have a partner. And in that case, it's more of we want to make sure that you have skin in the game. That's what that encouragement means. It doesn't mean that we'll throw a good proposal away. So at 2 equal proposals, we probably will look favorably towards the one that has a cost share. Likely that cost share will come because they have a business as a partner. But we will not throw a good solution away because there is not that cost share.
Right, for academic institutions that sometimes is challenging, but I understand how the industrial that the industrial side could help in that sense.
And I could add to that, because cost share is not mandatory, that would not be the deciding factor as to why we would discourage. So you are still allowed to propose regardless if you don't have that 20%. So I think we would be looking more towards the technology and the solution versus the cost share that you may be able to propose. Of course, we would like to see that 20%, but that won't be the sole reason why we would not select you.
You can think about creative ways. Like, we're not saying cash to pay scientists. Sam had a good idea, like, okay, maybe I'm going to provide 1,000 data sets or to the TA1B performer, which for you from an academic side, it literally costs you 0, but you can look at the market value of that particular data, and that has value. Like, you can arrange with a neuroendovascular neurosurgeon to acquire data sets. That is value, right? It doesn't mean cash to pay your postdoc.
Hi, everyone. My name is Louis Rogowski. I'm a senior research scientist at ARA. I have a couple of questions about TA2, specifically the composition of the microbots, because I know you want the electrical, mechanical, or hybrid between them. But if we had other components like chemical or nanoparticles, are you saying just don't use nanoparticles at all, or as long as we have one of those two components?
As long as you have one of the two other components, that's fine. It's just that the word microbots is used and the word nanobots is used to describe literally anything. People will call an antibody a nanobot, and we're like, we don't want an antibody, right? We want a thing that has a mechanical or electric component, and it might have all sorts of other bits of it, right? Like a single nanotube. It's not a robot.
No, of course.
But something that has nanotubes and also the little MEMS thingy, and the nanotubes move and do something. That's a microbot, right? So.
Sure. And just to expand on that, because the three things that were, discussed here was move, sense, and act. If one of those is only done by a chemical component, for example, is that acceptable? Okay, perfect. And then my final question is in relation to simulation and modeling, is there any interest in that occurring in technical area too, like concurrent to the physical prototypes that we've developed?
Yeah, this is not it's not mandated, but I can see that it could definitely be part of the development process, and maybe even a necessary part of the development process. So what you would do is you would include that as explicit sort of milestones and elements in your statement of work. If we looked at it and we're like, this is pretty ancillary, it does not actually seem required, you're just trying to fund some separate project.
Sure.
We might say probably don't do that if it seems core to and required for the development of your microbot. That seems reasonable.
Great. Thank you guys so much.
I think in general, there's been a lot of questions about what elements that are not required in the ISO can be included in your proposals. And I think the blanket answer, if there's a blanket answer for that, is make a good case for it. If you think it's necessary and useful and helpful to make your solution stronger, then by all means, they're not excluded.
And just to add to that, since many of you are not familiar with the ARPA model, we work very differently than NIH, for example. So once you submit something, it's not a take it or leave it, right? So there are negotiation processes at multiple stages. So for example, you may submit an abstract in which you say, we do ABC, and we go, okay, we encourage you, we really think A. So at that point, we will have a conversation between the solution summary and the proposal stage, and we say, hey, A and B, we feel it's great. You say we're gonna do C, like we really don't see the point. But since you're saying A and B, we really felt that D and E should be part of the process, so we would like you to consider that. Right, so that's one negotiation stage, and then after the full proposers say you were selected for award, there is another negotiation stage in which we go, hey, you said you're going to do X, Y, Z. We really do not like Z, cut Z. We don't want it, but we ask you to do this other thing which you didn't spell out that we felt is really necessary. So you need to build that into your work statement.
Hi there, Sean Taffler from Acoustic. Couple of quick questions on the stroke aspects of this. What's the minimum vessel size you're looking to target?
I would say based on the evidence, probably an M1 occlusion.
Dumb engineer.
Yeah, so like 2 millimeters.
Okay, or less if you can do it, I
Yeah,
guess.
yeah.
Yeah, so down to the smallest catheter you can get. Right. And for the delivery of embolic agents, is that for hemorrhagic stroke treatment, or is that just the idea that you could embolize anywhere else in the body?
I don't think we really flushed that out. I think it goes back to the first question. The ability to use this robot as a platform for many different things in neurovascular, even beyond neurovascular pathologies.
Okay.
But I mean, to answer directly, I think you should do that in the context of neuroendovascular procedures, again, with the thought that Adrian spelled out, that you may need to inject that anyways, right? So demonstrate that, and obviously there will be other applications, but there are enough risks in this program not to extend them unnecessarily.
Right, and so that brings me to my third question is, what's the possibility of applications that are not endovascular but achieve the same results as an endovascular procedure? For instance, histotripsy, delivery of ultrasound to lyse clots within the brain through the skull, no surgery, completely non-invasively. Would that be appropriate?
So we had received some of those, and we actually went back and forth among us. So I understand where you're coming from, and it can be a useful solution. There are also some inquiries about drugs that could potentially dissolve the clot, right? So they could be a solution to the fundamental problem. But they're really inconsistent with the way we had defined the program, right? So it's really meant as a robotic endovascular problem. Mm-hmm. That being said, we have means for the applicant, so our funding is given both top-down, bottoms up and top-down, right? So what you see now, it's top-down, in which we informed ourselves and we told you explicitly what we want. There is the bottoms up. We have the Mission Office Innovative Solution opening. So if you search for ARPA-H ISO, you will find 4 of them. for such a solution that you're proposed, like how about we target the clot through the ultrasound, that would be quite appropriate for health science futures. And I would definitely ask you to consider applying to that. So it is a valuable proposition. You have to think twice because you're breaking the clots and you're throwing clots in the whole brain, and we don't want that. It's a valid approach, but it really shouldn't be part of this program.
Okay, all right, thank you.
Thank you.
TA1 is fundamentally endovascular, and part of that is because it can also address other conditions outside of neurovascular too, cardiovascular, for example. And back to your question about embolization, one of the early thoughts was, what if during your procedure you perforate the vessel, you would have to address it somehow? So embolization sort of maybe was born out of that, but there are other applications for that as well. So we thought that would also be a very clinically valuable capability to have.
Hi, my name is Emily Shaw, and I've been in healthcare simulation for over 20 years. So I have had the pleasure of seeing this evolve to this point, and I'm very appreciative of the panel being here to answer these questions. I have 3 questions. The first is there a plan for partnering within government entities to ensure efficacy of the program in the field? I work with VA medical centers across the country. It's the #1 largest medical system throughout the country. 1/3 of all vets live in rural areas, so I hear your pain point about access to cath labs. The cath lab repository for outcomes data, called CART, has already recalled 3 medical devices and retroactively provided that information to FDA to recall those devices. So my question is there a plan to partner with SimVet and other entities that help to push those training programs out in tandem with preoperative planning to be able to tether to those databases and look at predictive validity? So what are we doing with simulation to better enable these procedures, and then to look at outcomes data and make this more of a standardized requirement across the board for medical device testing, starting with endovascular? So that's my first question.
I will throw this to Aldo. I'll give you like 30 seconds to think while I'm speaking. So we didn't immediately have a plan for that, but from what we're seeing, our programs tend to be quite visible, and we're sort of highly connected within the government. As you see, the 2 of us already work together, and as soon as it becomes evident that being in the same loop with other entities, reaching out is not a problem. So if at certain point during the program it becomes clear that we need to partner with somebody else, we will do that. It wasn't planned up front, but maybe Aldo has different thoughts.
No, I don't have a different answer. I think it's a little early for that type of approach in the real world. And I'm sure through ARPA-H Office of Commercialization, and once we are further down the path of actual products that can be tested in the clinics, then that's a reasonable approach. But I think at this moment it's premature.
May I proceed to my next question? Thank you. My next question is about mandating simulation as part of the roadmap for integration. Is there a plan to work with accrediting bodies for different physician groups, hospital systems, malpractice insurance companies, to look at a way to mandate this new tech across the board? That is something that I've seen a disparity with in the last 20 years, is that great programs come to fruition, and then the mandate plan and building it into accreditation and exposing residents early in their training, for instance, has been a real hurdle.
Wow, okay, I have to improvise on this one. So the one thing that we have, so on the one hand, like we all have to realize that this technology is like really out there, right? So I will be at ARPA-H for a little more than 3 years. But realistically speaking, this won't be in a clinic for a while, right? The reason we are commissioning that ELSI study that I had mentioned up front is just to understand what roadblocks this technology, if successful, will face, and try to address those roadblocks very early on. So I suspect that study, which will be done in the first year of the program, will likely come up with roadblocks of the form that you mentioned. And then we can start working with the relevant entities to try to plan for that wide-scale deployment. But, I mean, it remains to be seen which way we go, right?
No, I appreciate that.
Yeah,
Please
and
continue.
if I can add on a little bit to that too, ARPA-H is primarily focused on the development of moonshot technologies. And right now we're so nascent, we really have to, focus on de-risking the technology and demonstrating that there's something there before we start really moving into the more transition and adoption elements. But we need to be planning for that too. The way you framed it though, of having kind of a mandate to do this, that strays almost into a policy area, and that's something that ARPA-H doesn't work in, right? We can try to highlight the utility of something, right? But I don't think that the agency itself would be like trying to have a mandate put out there.
That's completely understandable. Thank you. My last question has to do with partnering with companies across the board in the world of simulation is quite challenging to get medical device companies to work collaboratively together and still protect their IP. So is there a specific language that we should focus on as a simulation company that works with 90% of endovascular companies to bring to the table our software solution in a way that we're able to protect our proprietary information or IP, but also to collaborate with med device companies? Is there any advice that you would give to our company?
Yeah, absolutely. I'll take a swing at it. I mean, so, disclosing what IP you have patents to and you will be claiming coming in at the beginning of the program is key. And as you're developing your teaming arrangement, creating a kind of IP management plan is a best practice, right? So what are you going to share? What elements of intellectual property that could be generated will each entity retain, right? Mm-hmm. We don't want to get to the end of a program and have a little bit of a knife fight over intellectual property, right? So working that out now as you're putting together a team is a best practice. And, I think if you're invited to the full proposal, incorporating, what elements you're planning to share from the simulation with other parties will be important and being specific about it. I hope that addresses the question.
Yeah, at least the tip of the iceberg.
Yeah.
I mean, bear in mind that we are requiring you to make this public, right? So at the end of the day, after the completion of the period of performance, anybody should be able to dump whatever robot files they have and be able to generate a simulation. So as you build your simulation package, you should consider that at the end of the day, you want to limit the amount of hand-holding that you as the developer needs to do for anybody else to be able to plug and play their data and still get the simulation that they need.
That makes sense. Thank you very much.
Hello, my name is Hong-Soo Choi from UMass Amherst, and I'm thrilled to ask more questions about this microbot. So I think my first question goes to Dr. Kurt Eggers. So for TA1A, so there's an answer already about opening and closing can be done by human. But I think for rear interventions, you need to have the guide wire reach the aorta, and from there we can push up like aspiration catheter or guide wire. So my question is, when do you consider the actual autonomous? So is it okay to have everything ready from the guiding catheter? Means guiding catheter maybe be able to insert by human, and then from there we can do robotic intervention.
I think the way we described it was from access, everywhere between access and closure is all autonomous.
Okay. Then the second question, if I work on Tier 1A, that means I already have technology for Tier 1B. Can I assume that? Or, for autonomous, we need surgical navigation. But Tier 1 and Tier 2, Tier 1A and Tier 1B kind of separated now. So if one team working on Tier 1, can we assume that we have complete satisfied Tier 1B technology that we can use?
Okay, I'll try to answer your question and let me know if it's diverging. We recognize that as TA1A teams are developing their systems, they would require simulation and data collection. Each team would have to do their own. And if the one robot succeeds and then the other efforts are sort of wasted, you might say. So it was decided to combine the simulation and data collection efforts into one solid, cohesive effort and have that TA1B interact with the TA1A teams.
So am I understanding correctly? So TA1A independently need to have like imaging and AI to navigate the robot autonomously.
So let me try. So I think so if you look at the metrics and milestones for TA1A, I'm not sure how exactly you can meet those if you don't have your internal navigation software, right?
Yeah.
So the attempt was for TA1B to bring everybody to a common denominator. I'm not seeing how you can develop a robot that would navigate without the simulation software of your own.
Yeah.
But we want to make sure that everybody's on the same scale. So the purpose of TA1B is to bring all the simulation software for each TA1A performers on the same denominator. But honestly, I don't think you can just develop the hardware and wait for TA1B.
So that's right. So I agree with that. So anyway, the TA1 team then have independent imaging system and AI for technology for surgical navigation.
Correct.
Okay, just one more question is for TA2, for example, if we have a robot can be manipulated by magnetic field with a magnetic particle conjugate with the cell and then delivered to somewhere and then use it for like a glioblastoma or neural connection, can be considered satisfy the requirement for microbot? Or I'm asking because it requires two kind of function.
Yeah, I think it kind of depends on like what the nature specifically is of the like mechanical component in motion, and also the exterior propulsion system has to be like reasonably small and practical, but that is potentially in bounds. It would depend on the specific detail.
Okay, thank you very much.
Hi, everyone. My name is Al Mashal, and I'm with Current Surgical. I had a question regarding TA2 with the microbots. I think one of the description elements said it needs to be able to do a biopsy, and if you're delivering a treatment into the biopsy and deliver the treatment, it can't just be one. Could you define what you mean by biopsy? Are you referring to it in the traditional sense of a biopsy where tissue has to get extracted, then be reviewed by a pathologist at some point, or any sort of sensing that could be a digital biopsy would also count as meeting that requirement?
So do you refer to a GI pill or what kind of microbot?
Sure, it could be GI, any sort of microbot. Let's say you have a microbot that can deliver some sort of, just make it up, ablation. But you say it needs to also do a biopsy. What do you mean by
no,
biopsy?
we were referring to a GI pill for that in particular, right? Because we've seen proposals come with us in the past where the thought is, well, we see a pill, we saw a polyp, we blast it. And that's never standard of care, right? Because you want to know what it was before you blast it, and ideally you want to confirm that you left everything clean behind. So in that respect, we were concerned about not ablating something that may or may not be cancer without knowing if it were cancer or knowing that you took it all out. So we're really looking for an end-to-end solution. Like If you need to ablate something that's not cancer, then there is no biopsy needed if you know for sure what you're ablating, right? I guess it can be a sort of pseudo biopsy in which you may have the optical means. So you demonstrate that you have an optical technology that gives you very high accuracy in determining whether that tissue is really what you want to ablate. You don't necessarily need to take cells out as long as you can confirm that that tissue is indeed what you had intended.
Okay, so just to confirm, when you say, let's say in that situation, a biopsy, it doesn't have to be a traditional biopsy. It could be an alternative way to do the biopsy.
As long as you demonstrate that that alternative approach has a very high correlation with what the pathology reads.
Got it. Okay, thank you. That's helpful. Appreciate it.
Hello, Anthony Samir from the Mass General Hospital. Two questions. The first pertaining to microbots. Would microbubbles under the control of ultrasound beams, in which the ultrasound beam and microbubble in combination provide sensing, and then more powerful ultrasound beams provide lysis, cavitation, and so on, in combination with the microbubbles, be considered microbots? Would that be within scope?
So hold on a second. The microbubble is one entity, right? And it's not mechanical and it's not electrical, electronic, right? So it's just one entity, and you provide ambulatory motion from outside. I don't see how you classify that as a microbot.
In the presence of mechanical energy, the bubbles oscillate, and in the presence of a lot of mechanical energy, they explode, delivering very local energy. So I guess the question is, it is acting mechanically locally, but initiated by the ultrasound beam, not by the microbubble itself.
Yeah, that's not what we had in mind.
Mm-hmm. Thank you. And then a separate question about the autonomous robot. Around the application of that technology is, at least in part, the notion that it would make the technology more widely available for stroke care. And part of that is the, I think, assumption that patients will not have easy access to neurocritical care immediately afterward, or even for guidance of reintervention if necessary. Are advanced forms of transcranial Doppler or other elements for kind of monitoring of these patients and guiding reintervention afterwards within scope?
Within the scope of TA1A?
Correct.
I mean, maybe as an adjunct to something that we had to neuroendovascular. Maybe, Kurt, do you want to comment on that?
Yeah, maybe as a way to augment the robot, the endovascular robot, or provide feedback on where the catheter is, et cetera, but not as, like you said, not as an independent part of not as the endovascular robotic itself, but as an adjunct potentially for navigation guidance.
Does the application need to be intra-procedural, or is a monitoring application afterwards as a guide to reintervention acceptable? For example, providing similar information to cerebral perfusion CT, but more on a kind of ongoing basis.
Maybe you can answer that. I would probably say no to that, right?
Yeah, so bear in mind that this was meant as an end-to-end solution, right? So I see, you need, as part of TA1A, you need to validate that you had reperfused. And at that point, we would close the procedure, right? And again, if you see confirmation is pretty easily done by fluoroscopy, so I don't know why you need the ultrasound in that case. But again, we're not spelling out every part of the procedure. So if you can make a case that approach would be safer or better or faster or whatever than using fluoroscopy, we're not necessarily opposed to it, but we're not doing novelty for the sake of novelty.
Thank you.
Hello again. Just a quick follow-up question on technical area 2 regarding external power generation for these microbots, because you guys specified this can't be any bigger than a cart. And I'm not necessarily asking for the dimensions of the cart, but is the intent of that, it just has to be a portable system that somebody can confidently push around, even though it may be slightly heavy-ish. So
That's correct. We would prefer something under, 800 pounds. But again, the guiding principle of TA2 is that you have to, the procedure that you do in that way has to be easier done in that way than it would be otherwise, right? So again, we're not funding novelty for the sake of novelty. We want to see procedures being easier, simpler, more accessible than they are done right now. So you have to question, is the workflow that I'm proposing making the procedure simpler? Can it be done by somebody less skilled? And if the answer is yes, and presumably it's not going to be a factor of 10 more expensive, then we're interested, right? But we're not interested in something that's just cool.
no, of course. I was just thinking, like, if you guys wanted something really small or something, how do you?
I mean, a cart is a cart is a cart, right?
Yeah, of course. Great, thank you.
Hi, my name is Yoonho Kim from a company called Meganendo based in Boston. I have a couple of questions. The first one is in TA1A, it is the system integration with the imaging system is considered to be essential in the program. And that's very understandable because you need to control the C arm of the table on demand. But technical feasibility is just obvious. It's possible. But it's all about whether the imaging companies, they're only handful of, which in working in this neurovascular space, allowing you to be able to do that or not. And I was wondering whether you can elaborate a little bit on how you envision this system integration with the imaging system, whether you are looking for some bypass solution that doesn't require any communication with the C-arm directly, or you imagine that the proposals will need to partner with the imaging companies to be able to do that.
I worked for 20 years for GE. I don't know if all companies work the same, but we used to have collaborative research agreements with scientists, and we would provide them with access to our software for development, just like the ones mentioned. So no, we're really not interested in hacks. I mean, I don't have a sense of how collaborative the companies are. I think traditionally in the imaging space where I had spent most of my career, they were pretty open, understanding that in the end, it's in their best interest to, have broadened their market, right? If the companies don't, I don't want to hack a system because in the end, that's never going to be a product. So your best solution would be to make friends with those companies and make sure that they support you in some way or another. If that's utterly impossible, again, I don't know, maybe Kurt, do you have any insight on how collaborative these companies would be? I mean, we have some leverage. In the past, we have been able to reach out and accomplish things that the proposers themselves couldn't, not the proposers, the people under award. They couldn't accomplish. We stepped in and we convinced people to help us. But we would prefer you do that on your own.
Right, but given the fact that the number of imaging companies, the major imaging manufacturers are really small, let's say 3 to 4, then are they allowed to partner with multiple companies if they just offer that connectivity or communication with the Andrew suite?
Yes. So I don't think we should limit the number of performers. I mean, in the end, it's not like we're going to award 20 awards, right? So I think it will trickle down, and we understand that's a crucial part of each and every proposal. So it's unlikely that we'll limit that. Also, I don't see them needing to do a ridiculous amount of work either, because the subroutines to control the C-arms are there. They just have to give you access to them. So maybe some support person, but I don't see them spending, millions of dollars for that to happen.
Right. So I was wanted to mention or point out that 4 of the 2 companies have already building their own robotics program, so they might be in conflict of interest if they were to partner with other companies. So the remaining options will be very limited. So I just wanted to point that out. My second question was, in terms of teleoperation requirements, there are some words like connectivity, latency, these kind of requirements, but those are typically the case for long distance remote procedures between hospitals, between cities. But are we required to demonstrate that? Or if simple teleoperation, like hardwired connection, so the physicians can do the control with a joystick outside the cath lab in the control room, that will be sufficient. Just want some clarification on that.
So one of the early milestones for TA1A is teleoperation, and that wasn't specified as remote teleoperation. It can be if you think that's a better solution, but it's specified as teleoperation.
Right, so if it's a hardwired, so latency or connectivity is not an issue, then we don't need to demonstrate all those other requirements out of the scope, right?
Right.
Okay. My third.
It depends on what, I guess, hardwired. To what extent? Like, are you using a local hospital or a local facility network? Are you using straight?
No, what I mean is the joystick controller or the interface directly wired to the system, so there's no latency at all. In not wireless, control.
So I agree that we did not specify that. I don't think that's where we want to go, right? So if you think about the evolution of a system, again, so the way we thought about it is, yes, in the first implementation, you develop the system, a surgeon will always be there, it will be done with autonomy, but then a surgeon steps in. Like, if this works, the clinical trial is successful, then you can foresee one endovascular surgeon being remote and overseeing the operation of multiple such robots. So really, you need remote operation.
Long distance remote operations?
I mean, at least over Wi-Fi. Like, it can be in the same hospital, but not wired.
Okay. Thanks. My last question is regarding the FDA collaboration, because the neurovascular division who are reviewing those devices, given the fact that there is no FDA-cleared neurovascular robotic systems as of today, they are concerned about, let's say, haptic feedback, lack of haptic feedback. But I was wondering how they, the folks who are actually reviewing those devices, are involved in this development program. So while building on the Phantom model, for example, because I was told today that we'll be given a 3D model of the Phantom so that we can 3D print ourselves to build a model to test. But it's not that simple, because the mechanical properties, surface properties, these are very important. So I was wondering how deeply this FDA neurovascular people will be involved during this program.
So the goal of our involvement is to develop these methodologies, phantoms, and other testing methods, so that when the review offices receive packages from these devices, the data that is obtained through the bench testing and other methods can reduce the size or the burden on large clinical trials. Of course, they will need to demonstrate that. But the more data we have, the more confidence we have on the devices through other means, reduces the size and effort on the clinical side. Our current thinking is that we will include their direct participation in the quarterly meetings, so that, and we'll probably have more internal meetings as well, so that they can advise on the work that we will be doing and how the progress of the performers as well. That doesn't necessarily answer the question, what's going to happen when the files come, right? We can't answer that question because until that moment. But what we can do is advance the methodologies so that we gain confidence on the data that comes from them, that they include the right patient variability or task variability that typically these devices will be asked to provide, the submissions, and other factors that might be important. You mentioned a number of them. So that's kind of the level of involvement that we want. And as you know, that's an initial setup. I suspect that as we get closer and closer to the more final stages of the programs, and as you, the performers, get closer to a product, then there may be other opportunities that, and this may include, the traditional Q subs that can be set up, as you know, by anybody to discuss details of the future applications.
Great. Thank you so much.
Shall I go next? Thank you. Johnny Bittio from WPI. I was wondering if one was to participate to TA1A with different platforms as a proposer. Is that okay? Or is there a problem there?
So we said that we are allowing each team to have up to three. We don't want to see one name more than 3 times, and you can be prime once and sub twice or sub 3 times, right? So you can have your name can be part of 2 different proposals for TA1A, but you can only prime one.
Oh, okay. Thank you.
Madison Clark Turner from Charles River Analytics. I have 2 quick questions. The first is you mentioned with regards to the encourage-discourage decision that you would be able to provide essentially a selection of options, and you would tailor your encouragement or discouragement to individual options. Is that accurate? And second, is there a limitation? Or I guess what I'm trying to get here is the detail in that encourage-discourage decision. Is it just a yes/no versus a more detailed response?
So in the solution summary stage is when we would decide if we would encourage or discourage based off of you meeting the ISO requirements. If you are discouraged, you're still allowed to submit a full proposal. But of course, there's a risk that comes with that, right? Because we have discouraged you at the solution summary phase, and we realize that there's a time and money that goes into a full proposal. But you would still be able to move forward with submitting a full proposal.
But I think the question was slightly different. So you were asking if you're going to provide you feedback, such as you can decide, well, okay, they didn't understand that, and I'm going to go ahead and still submit because clearly what was listed as a weakness is really not. Is that what you meant?
Perhaps I should clarify. If in addition to the regular solution, we were in addition to that suggesting a perhaps swap C limited approach or a connection limited approach as an optional add-on to that capability, would we receive encouragement or discouragement for those extensions beyond the base capability?
So as I had mentioned before, there are different negotiations, but feedback stages, right? So if you in the initial submission, you have A plus B plus C, then if you are encouraged, then we will spell out what we want and what we don't want.
Understood. Okay, thank you.
If you are discouraged, it really depends. So it depends how many, I had been a scientific review officer for 3 years, so I do respect the review process. I had been a PI for 20 years, so I kind of would like to have feedback. And we generally do provide it. However, the extent of that feedback depends on how many solution summaries we get. Like for the initial program, we had well in excess of 100. And as you can imagine, writing a comprehensive feedback letter takes a whole lot more. I cannot take 4 weeks to do it because you don't have time to write the proposal. So the extent of the feedback will depend on the ratio of how much we receive to how much time we have.
Thank you. I appreciate that. My follow-up question, my secondary question is, with the T1B timing, presumably that's being developed in parallel to the T1A effort. will there be a standard for our capability to communicate with that simulation? And if not, an estimate on when that will be delivered? Would we be expected to develop a simulator ourselves for the T1A teams?
We're starting to see how much coordination would be useful and productive and necessary between TA1A and TA1B, and that's the vision for that. There are specific time points and deliverables in the ISO for when things should be available, things should be delivered, but you're also encouraged to have more iteration. And TA1B also has to work with the FDA to keep the FDA apprised of the developments there too.
Okay, thank you.
And to add on, I think I answered this question before. Like we fully expect the A1 performers to have their own simulation environment. It's just not going to work if they wait for you to develop that. There will be, as Sam was mentioning, there will be a kickoff at which all the teams will be together. It will be in everybody's best interest for the TA1 A and B teams to work together. And so, yes.
Noted. Thank you very much.
All right, good afternoon. My name is Xiangling Jian from North Carolina State University. My question is related with the previous one. I heard, ultrasound-based microbubble-mediated sonothermolysis was not considered. Could you please clarify whether that includes the ultrasound catheter-based endoscopic ultrasound thermolysis? Also, that's not something what you're thinking about, because we're not talking about totally replace the mechanical thrombectomy. That can be a very valuable addition to existing thrombectomy technique.
Yeah, I think if it's something delivered by a robot, then that's fully within the scope. I think if it's independent, then no.
Yeah, it will be robot. Yeah.
Yeah.
Thank you. The other quick question is about, do you have any suggestion if I have two contrasts? One is FDA approved, the other is not approved yet, but the other one shows more promising results. Do you have any suggestion which one I should go with?
I think Aldo's not allowed to have these kinds of suggestions.
This is an investigational use.
Yeah, so whatever more promising we should go with.
Yeah,
right?
I think you should target whatever works better for the technology.
Yeah.
You might want to do a bridge study comparing the difference between the two agents and see how much of a penalty that is, but you should focus on the best product development.
Yeah, clear. Thank you.
I mean, but you also need, again, so bear in mind the guiding light is we want, at the end of the day, an end-to-end solution. And you need to understand the roadblocks towards having that particular agent become a product, right? So there are so many risks to this program. We don't want to consciously pile up more risks. Right. So if that specific agent that you refer to is on some path to productization anyways, and it's only a few years until that becomes product, then I wouldn't be worried. But if you rely on TBD to pick it up and take it to clinical trials, then that compounds the risk in a somewhat unnecessary manner.
Gotcha. Thank you.
Yeah, one thing I will say is we have had performers who are for Alana's other program where their technology does rely on a novel contrast agent, right? And that's kind of a predicate for their overall success, and it's something they had to work on early for getting through FDA. As you're structuring your proposal, though, it sounds like you might have two options, right? And you can have a backup. There can be redundancy, potentially, depending on how it changes your technical approach.
Yeah, we have the other one, actually, paperwork is ongoing, so probably good to go. Thank you.
Okay, so Paul is going to read the questions, and one of us will answer them.
First question is, are you expecting an LC breakdown in the submission, or is LC done by ARPA-H? That's ethics, legal, and social implications.
So we will be doing it, and we will pass the results to to whoever is under award. So I feel it would be wasted effort because we probably will do it more carefully than anybody can because we have the time and resources. So we'll give the results out. You don't have to worry about it.
Right, but that is an interesting aspect of this program. Okay, next question. I think this is similar to what has been asked. why is embolization mentioned if the primary goal is to autonomously revascularize?
I think we answered that already.
Okay, thank you. And then next question. Does a GI pill robot that can operate under its own power in a telemedicine setting, doing biopsy or other interventions, look like a TA2 fit?
Does this one imply that it's teleoperating or is it autonomous? Or is
A GI pill robot that operates under its own power.
I think I think that's in scope, right? Like because
Yeah, that sounds reasonable. We just did not want GI pills to ablate blindly. So that we don't want a GI pill that images because they're already products, and better imaging is not the solution to anything in the GI pill space. And we did not want GI robots that ablate blindly, because that's not done in the clinic. You don't know what you ablate and you might mask a problem. But GI pills that biopsy are fair game.
Next question, are foreign partners allowed?
Yes, foreign partners are allowed to propose. We do encourage that you propose with a U.S. partner. The only exception to that rule is that we would not consider awardees that are part of a foreign country of concern.
All right, this is a clarification on submissions. Are teams to propose TA1A and TA2, TA1B and TA2, or only to one? So I think this is a clarification.
There is the master slide that Gina had, which basically highlights the fact that we want a grand total of 3. You can be prime on 1 and sub on 2, or you can be sub on 3. So you can propose to more than one TA. At the solution summary stage, you can propose both to TA1A and B if you want. But if you are selected for and encouraged to submit a full proposal, you will have to choose. We don't want any mingling between TA1A and TA1B.
yeah, just also clarification, that's a max of 3. If you want to submit 1, that's good. It's perfectly fine. And yes, you do not have to apply to multiple TAs. I think that's what the person was asking. If you are a microbot developer, apply to the microbot one. Don't apply to the other ones.
Okay. Next question, is the restriction for more than one team from an entity not being allowed to apply to TA1A and B, as determined by a single UEI, applicable to distinct labs with distinct technology coming from the same university?
No, so as we had specified, the definition of the team is, as it pertains to academia, one team is essentially one lab PI. So if there are 2 independent labs with 2 independent PIs in the same university, those are considered distinct teams. And we can have one team apply for TA1A as prime and the other as TA1B as prime.
Okay, I think some of these questions are starting to repeat, so I'll do a little bit of curation. Sorry if I skip your question. If it doesn't get answered, ask us later offline. Next question, would a proposed solution that requires substantial development of a new imaging capability, for example, 3D imaging, be considered responsive, particularly if the effort is roughly balanced between imaging system and robotic platform development?
Well, I mean, it depends on the exact solution, right? This is a robotic program. This isn't an imaging program. I can see how there may be some need for optimization, but half and half sort of seems not what we had intended. So we understand that as both part of TA1A and B, you may need to do some imaging development or optimization, but half of the entire program does not seem appropriate.
Next question, for TA2 microbots, does sensing need to be on board or can it be off board, such as with external visualization systems?
I mean, we said the microbot has to sense. It's kind of difficult.
I'm not sure how external visualization sees inside the body.
Yeah. Honestly, hard to say. It would really depend what you mean by external sensing here. Like we do ask that the microbot senses, but then again, we do allow for some amount of external aids.
So we are okay if, for example, you have ultrasound and you need to visualize a microbot, that's fine, but it's not clear what sensing means. So, I mean, at the end of the day, we cannot know without seeing the solution summary, and the solution summary is 4 pages long. So if in doubt, I just submit.
I think the guiding principle is microbots. So if you have such an onerous large imaging system that defeats the purpose of having a microbot, then that's not what's envisioned. All right, next question. For TA2, can you expand a little bit more on the restrictions for microbot propulsion? Would a microbot that relies on passive propulsion only be allowed? Introduction placement devices allowed as part of the propulsion strategy? For example, catheter device or flexible tether that helps the robot travel part or all the way to the target site, but doesn't remain in place after introduction.
Well, again, the guiding principle is that it should be simpler than what we do right now, right? So if you're just going to use a catheter to put a robot where it needs to be, that's not a robot, that's a catheter, right? But high level, for example, if you can make a procedure simpler by placing it somewhere that's easy to place, that's close to the target, and then the robot navigates the last part on its own, maybe it's not trivial to place it surgically. Like if you need to reach certain regions of the brain, and it's easier for the microbot to just navigate the last part where a catheter couldn't be placed, I think that's fair game. But if you're just going to put something with a catheter somewhere, I don't see how that can be called a microbot.
Okay, next question. For academic institutions, will student tuition be permissible for these OTs, or is the intent to run these projects largely or exclusively with postdocs?
No, we do negotiate tuition remission. You're okay with that, right? Yeah, so it's something that we typically negotiate. It's not always guaranteed. It depends on what is being proposed and the personnel that are being proposed from that university.
But we do have other transactions with plenty of students participating and being paid from the OT, so it's not a limitation.
For approaches that don't rely primarily on fluoroscopy, can you say more about what is or isn't acceptable for additional imaging tracking equipment inside or around the patient? Is it reasonable to assume that anything that rolls in on a cart is okay?
The main goal of getting rid of fluoroscopy is getting rid of radiation to the physician and the patient, right? And the C-arm is not negligible in size. So if you get rid of fluoroscopy, then I think it's fair to replace it with something of a, cart size, right? So you have the C-arm in the OR anyways clogging the space. Maybe I'll let Kurt comment.
I mean, initially you're going to need some kind of visualization for backup, like physicians to verify location and whatnot. But I think ultimately the goal is to get away from that for those reasons. But I think initially you're going to need some kind of visual feedback so a human can actually respond to what's going on with the catheter.
So you're saying for development purposes you need both, but ideally towards the end, if you can develop a guiding system that doesn't rely on that and you demonstrate that it's equivalent, then the ultimate solution is fine. That will take some real estate in the OR.
Yeah, I think it's reasonable.
Yes.
All right, next question. Confirming, are clot removal and vascular embolization intended to be part of the automation, or is the automation limited to navigation only?
It's intended to be all. They are intended to be part of the automatic solution. And so that includes logic to figure out, for example, that you've made contact with the clot so that you can now get it out.
I think with aneurysms and other things you're embolizing is going to be much more complicated than that. So there's going to be a lot of human input. But I think for the initial thrombectomy indication, it's going to be all autonomous.
Paul, I think we have time for one more question. Unless that's it.
Oh,
Sweet.
we're done. Thank you all, and looking forward to reading the great solution summaries.